को अपनी रोशनी से प्रकाशमान कर रहे इस विश्वविद्यालय की शुरुआत साल 1966 में हुई थी। शिक्षा जगत के मानचित्र पर आज यह विश्वविद्यालय प्रदेश की 11 जनपदों में लाखों विद्यार्थियों के भविष्य का निर्माण कर रहा है। गांव कस्बों से लेकर आसपास के शहरों तक के 600 से अधिक कॉलेज विश्वविद्यालय परिवार से संबद्ध होने के कारण इसका हिस्सा बने हुए हैं। अकेले विश्वविद्यालय परिसर में ही कला, विज्ञान, वाणिज्य, विधि, अभियांत्रिकी, चिकित्सा, समाज विज्ञान, साहित्य, पत्रकारिता, प्रबंधन, ललित कला समेत अनेक पाठ्यक्रम स्नातक एवं परस्नातक स्तर पर संचालित किए जा रहे हैं। नए पाठ्यक्रमों का सृजन, नवीन विचारों का संग्रहण तथा तकनीक व आधुनिकता के सभी सफल प्रयास विश्वविद्यालय के एकेडमिक जीवन शैली का हिस्सा बने हुए हैं। इस विश्वविद्यालय से उपाधि अर्जित करने के पश्चात यहाँ के उपाधि धारक समाज में अपनी विशिष्ट पहचान के साथ संस्थान का नाम भी रोशन कर रहे हैं। सार्वभौमिक उत्थान के लिए नित नए साझा प्रयास, जी तोड़ मेहनत एवं नवाचार के लिए प्रतिबद्ध विश्वविद्यालय को वर्तमान स्वरूप माननीय कुलपति प्रोफेसर विनय कुमार पाठक की सोच का परिणाम है, जिन्होंने अपनी दूरदृष्टि, कुशल नेतृत्व क्षमता एवं विचारशीलता से विश्वविद्यालय के ऐतिहासिक स्वरूप को तो बनाए रखा ही, साथ ही उसे आधुनिक तकनीक, कौशल के समन्वय से जोड़ दिया है। समाजोपयोगी पहल, जन जागरण के विषयों के साथ साथ विश्वविद्यालय ने जन सरोकार के अनगिनत विषयों पर जागरूकता अभियान, कार्यक्रम, संगोष्ठी, नवाचार के माध्यम से अपनी महती भूमिका निभाई है। एक किराए के भवन से अपना सफर शुरू करने वाला यह विश्वविद्यालय अपने शैशव काल से मुक्त होकर नित नए आसमान को छूने के लिए प्रतिबद्ध है। हम सभी के लिए यह गौरव का क्षण है कि ज्ञान दर्शन की इस विरासत के 57वें स्थापना दिवस पर हम इसके साक्षी बन रहे हैं। इस उत्सव के रंग में हम विश्वविद्यालय परिवार के उन सभी शिल्पकारों को प्रणाम करते हैं जिनके अथक प्रयास से यह आज अपनी आभा बिखेर रहा है। दुनिया भर में
Good morning to all of you. It is an absolute honor and privilege to stand before you. I, Dr. Shweta, with my team, on the behalf of the School of Pharmaceutical Sciences, Chhatrapati Shahu Maharaj University, welcome all of you to this prestigious international conference on pharmaceutical frontiers, exploring future trends and technologies, pharmaceutical innovations for the new era of healthcare 2025. This event marks not just a gathering of great minds, but a convergence of ideas, innovation and inspiration. We extend our heartfelt gratitude to our esteemed guests and speakers who have taken their valuable time to share their insight with us. So before we move ahead for the inaugural ceremony, in the meantime, we feel privileged to welcome Dr. Abhay Chauhan, who is joining us virtually from Wisconsin, USA. So, I would like to tell you that this arrangement is all because of the difference in the time zone. So, before we start, I would like to give a brief introduction about Dr. Abhay Chauhan, sir. Dr. Abhay Chauhan is an expert in nanotechnology-based drug delivery system with main research focus on Dendrimer nanotechnology. Dendrimers are widely recognized as members of the fourth major architectural class of polymers after linear, cross-link and branch architectural types. He is working towards preparing Dendrimer-based nano device for the agnostic applications. He has carried out detailed pioneering work on nano device, establishing Dendrimers for formulation development. Stability and solubility enhancement. Dendrimer-based transdermal nano formulations, multiple drug delivery, reduction of drug toxicity, nose to brain delivery, and multifunctional dendrimers for drug targeting. He has successfully developed Dendrimers formulations for pharmaceutical, in vitro diagnostics, personal care, and agriculture applications. His innovative work is published through reputed journals and patents. He teaches various pharmaceutical subjects and mentors students for the undergraduate, professional, and graduate programs. Dr. Chauhan served as a reviewer for various renowned journals and grant reviewer for NIH and other international agencies. He is involved in creating nanotechnology awareness globally and currently working on understanding critical nanoscale design parameters in Dendrimer nanoparticle for drug delivery applications. Dr. Chauhan is also the co-founder and chairman of Amrit LLC to commercialize Dendrimer technology. Amrit in the phase 1 and phase 2 human. So I welcome you, sir. Over to you, sir. Thank you.
Hi. Hello everyone. Can you guys hear me? Am I audible?
Yes, sir.
Okay. Thanks for the introduction. And should I start my talk or we are waiting for something?
So please start. We are ready for.
And the slides. How should I share the slides? Should I share my screen here?
Technical persons will help you. Sir, you just share your screen. So please share your screen.
Let me get the right thing. Can you see my screen?
Yes, sir. Yes, we can see it.
Okay. So,
Sir, I request you to please, sir, make it a full screen.
Is it not full? It's
No,
full
sir,
from
it's
my
not
side.
full.
It is the position mode.
Okay, sir, fine.
Tell me what should I change? Like this you want?
No,
This
sir.
is not condition 4.
Sir, please maximize it. Means full screen. Will it be possible to
Yes,
make
full.
it? Is it full? From your side, is it full? Showing full screen?
Yeah, it's from my side.
Okay, so.
Let's start it.
Okay, sir, let it be, sir. You can carry, sir. Please start.
I was trying to see that, what do you guys see that side?
Front page, sir. First slide. That is Dendrimer nanoparticles and commercialization.
So yeah, from my side it's full here.
Okay, sir. Okay, sir, it's fine.
Not full. Trying to see something that we done with each side of the No, I think that's what it is, how it's showing here. Okay. So, first of all, thanks for inviting for this presentation. And my name is Abhay, and I'm faculty at Medical College of Wisconsin. My research is basically on Dendrimer nanomaterial. And I'll talk about Dendrimers first, and also my efforts to bring the product in the market based on Dendrimer nanomaterial. So, before, I don't know what's my audience, are they undergrad or PhD students, masters? But basically, whenever you develop or work on any drug delivery system, first thing you need to understand the journey of the drug. So drug is not a, it's a kind of mostly a dead organic molecule, non-organic molecule, which has to move inside the body. people always say that rocket science is complex. Okay, but mostly when rocket scientists develop something for the make a rocket go to space, mostly they have good idea for the space. Unfortunately, we are still not that much aware around the anatomy and the physiology of the body. The knowledge is still very limited. So we need to understand what happens to drug when it goes inside the body. The journey of the drug is very important. Until you know the journey of the drug, you cannot develop a system to control its movement. So for example, you take something orally, like a tablet, what happens to tablet? This question should be with all of you in your undergrad or the grad or doing PhD. That's what happens to that thing. A tablet, for example, goes in the stomach or in small intestine, breaks into particles, this disintegration, then dissolves, dissolution. Whatsoever dissolves must permeate to the GI membrane through various mechanisms. From there, to portal system, it goes to the liver, gets metabolized, and then the metabolized and non-metabolized blood comes to blood. From blood, it goes all around the body. Again, from there, it has to leave blood between the endothelial cells. From there, it comes to the interstitial fluid, where all the organs are bathing, literally hanging there. Drug has to basically swim through that whole interstitial fluid. Then they come in contact with tissue, and then there they have to cross the cell membrane. Then they come to cytosol, and there they have to swim through it, and then nuclear membrane, then goes inside the liver. So, literally in non-living organic molecule has to cross these many barriers. And that's not an easy thing to do because there are many factors besides movement of drug inside the body. So first, we need to understand what are the barriers for movement of a drug inside the body. Once we understand and where drug has to go, it's like if you move from place A to place B, the city area, today, you need to know how you reach the area, right? And you can walk, right? If there's a park, or you can drive the car, or now you can have GPS in the car, or your phone, Google Maps, right? Tells you to where to go, right? That's called targeting. Nowadays, for example, the Tesla is making self-driving car. I myself have a Tesla car and it drives nowadays. I used to sit there. The thing is that similar things can be done at very small level, nano level, for delivering drug inside the body. And that's very important. So, before you, and there are various ways, means there's no one way to move drug from place A to place B. There are various delivery systems. Many of you know polymer-based delivery system, or ribosome-based delivery system, nanoparticle-based, various micelle-based. There are various kinds of delivery systems. I work with one kind of delivery system. Right? Actually, my whole, from my master's to PhD to my job, from last 25 years, that's what I'm doing basically. So I work with something called dendrimers. Dendrimers are basically the fourth class of polymer. The initial polymer, like a linear polymer, were there in the 1930s, gives us nylon and plexiglass. Then they said, okay, fine, can't we take polymer and cross-link it to make it a complex structure? That gives us rubbers and epoxies. Then they make branched polymers that gave us polythene, which is used a lot nowadays for everything, carrying everything. Then, the late 80s, people thought that, okay, fine, can we make these polymers in defined structure? Like the current way of making polymer was that you take a monomer and polymerize it, and then you get some polymer. Right? Those are not very defined structures. But can we make precise layer by layer defined structure? And that led to dendrimers. So dendrimers are basically the fourth class of polymers. And this is a typical schematic diagram of dendrimers. So basically, let's see the So you see that here this is called core. There's a core molecule. This core should be at least 3 times. I should have a hello? Lights down.
No, sir. Your slide is not. Macromolecular architecture.
Okay, this slide you can see, right? Okay, let me move one more slide and you will tell me. Is it moving now?
Yes, sir.
Maybe I should not use the presentation mode because I have no idea.
Now it is moving, sir.
Okay, let me try presentation mode. Is it moving now? Is it moving?
No, sir. It is moving, but it is not in presentation mode. I think so.
Okay, so which way you want? Which way you want? If you like, sorry. So, like this way?
No, sir.
Can you see now? Just use the tab or different way.
Now it's okay, sir. So you can use your touchpad or mouse for just moving the slides every time. Yes, sir. Like this, sir. Yeah.
It's moving now?
Yeah, sir. Yeah, now it's moving.
So I will do one thing. I will not use the presentation mode because the reason something is happening, right?
Oh, okay, sir.
I will do like this.
Fine, sir.
Okay. So I'll go back. Sorry for I have no idea why it's happening. So I was explaining this slide before that there was a linear polymer, cross-linked and branch polymer, and the fourth class is spherical polymers called dendrimer. So these are dendrimers. You can see here. These are the it has a core. You can see the core molecule here, and then the core molecule branching comes out. Can you see a mouse moving or no? Can you see the mouse moving?
Cursor, yes, sir.
Okay.
Moving.
So, can you see the mouse moving on the dendrimer structure?
Yes, sir. We can see that clearly.
Perfect. Thank you. So you can see it has a core molecule, right? And then it branches out. So each time you add a layer over this, so generation 1, then you add next generation 2, keep adding like that. After some time, it gets a spherical structure. So dendrimer are a precise architecture at nanoscale, and the core, and you see that there are spaces in between that helps to entrap any molecule. It could be drug molecule, it could be imaging molecule, and the outside it creates polyvalency. So meaning Z outside here, this creates polyvalency. So this can be used to attach multiple drug molecule, or non-covalent and covalent interaction, or imaging molecule. You can attach multiple things here to create some kind of nanoparticle. So you can see that. Can you see my slide moving now? Oh yeah. So you can see that each generation, you keep adding. After some time, it takes a spherical shape. The generation 4 to 6 are mainly used for delivering drugs. So drug can be entrapped. Generation 7 onwards can be used for gene delivery. Because a lot of charges happen on the dendrimer surface, it can be used to attach any kind of gene, DNA, RNA, mRNA, etc. But we basically, what percentage was for generation 4 to 6. These dendrimers are very precise structures, unlike other polymers. So if you do any kind of a size exclusion chromatography for GPC, for proceed to the molecular weight, you can see a typical polymer peak comes very broad. But you see each generation dendrimer peak comes very sharp. It shows that these are very precise structures, unlike other polymers. You can also see these dendrimers with the TEM transmission electron microscopy to show that they are very homogeneous. So they are very precise and homogeneous, unlike the typical polymer. Like many years back, I proposed this theory of critical nanoscale design parameters. What you have to understand is that making anything is not difficult, but making something at nanoscale is not easy. And nanoscale is important because all the building block of life, from DNA to RNA to protein or early evidence of life to viruses, are all in the nano range. So living body, our body is made of nanoparticles. So in a sense that we need to understand how nanomaterial behave. So that to deliver drug also, we need to have something at nano range. For that, we have to understand size, shape, chemistry, flexibility, architecture, and elemental composition. These are the parameters called critical nanoscale design parameters in Dendrimer or others. Dendrimer has an advantage of, basically the main advantage is that it has a polyvalency to see multiple groups outside. So you can attach multiple drug or a medium drug, emitting molecule and targeting molecule all in a single system. Whereas this and then you can trap drug inside. So it works both ways. So you can have a combination approach where you can trap drug physically, non-covalent way. That way no covalent bond is being used to attach the drug. That way the release of drug is easier. The second approach is nanoconstruct approach, where you can attach drug or any other moiety chemical. That's a nanoconstruct. The advantage of Dendrimer over others is that it's a multifunctional agent. You know, we all had 15 years back some phone or some form of GPS or camera or some various things, right? What does the smartphone do? It provides one platform for multiple applications, which is an app. So Dendrimer has that capacity in drug delivery arena where multiple application can be designed in Dendrimer architecture. So that kind of advantage over other deliveries. I'll give you a few examples, simple examples of Dendrimer which I have proven. So first thing we have shown many years back for PhD that these Dendrimers can be used as solubility enhancers. Basically, a drug can be physically trapped in the void spaces or by hydrogen bonding or charge interaction, hydrophobic drug. And Dendrimer's outside surface is water soluble, it increases solubility. And I think in 2003, mine was the first paper to explain the whole solubility phenomena. Somebody else published before, a short bit before me, regarding the solubility property. And we have shown that how Dendrimer helps in enhancing solubility. Similarly, we have shown that by changing the surface of Dendrimer, from amine to fat to petone to aminoethanol to carboxylate, succinic acid, and every possible surfaces, and each generation, generation 2, generation 3, generation 4, you can see that. And we have seen that its entrapment depends upon what kind of drug you pick and what kind of Dendrimer surface it has, and also the higher generation Dendrimers have a higher control. So we have shown all those things. And we have shown that once the drug is entrapped in Dendrimer, we get higher resolution and controlled release. And that basically leads to higher bioavailability also. I was the first one to show that these Dendrimers can be used for transdermal permeation enhancer. So we have entrapped drug and then with formulation in a patch, and we applied on the rat skin, and we have shown that we were able to increase the for the transdermal permeation of endomethacin, and we have done the whole study, so it was published on the back. Similarly, we have shown that we can deliver drug into brain through nose. So, delivering drug in the brain is a very complex thing because brain has blood-brain barrier. It's basically endothelial cells with a strong astrocytes. So they do not allow anything to pass through. So difficult to deliver drug. So what we have done is that we all have olfactory nerve coming from here to nose area, you can smell. So that area is a leaky area in blood-brain barrier. So the concept was there before that something can be used like that to enter brain area through nose, and we have used Dendrimers for that purpose. And we have shown that, so we can herbal drug because that way very fast it shows up some kind of pharmacological effect on the animal model. And we have shown that we can trap herbal in Dendrimer and successfully delivered into brain through nose, and we could achieve a similar result as the IP at 6 times over consumption. So that paper was published and accepted one of the best editor's choice of the ACS. And similarly, we have working on the concept of hybrid, like a next generation Dendrimer, where we can take multiple Dendrimers and bring them together to increase the functions there or to achieve better delivery targeting. And we published many papers on that, and we have shown that we can deliver docetaxel by using hybrid Dendrimer and increase the oral viability. Similarly, we created multifunctional. So in case of cancer and other stuff, what happens that, for example, brain tumor, which I worked on, when cancer grows in brain, it needs a lot of iron to grow. For that, they overexpress certain receptors, like a transferrin receptors or mannose receptor. So what we do that, we try to attach the ligands for those receptors on Dendrimer and then trap the drug in Dendrimer, hoping those receptors will stick to overexpress receptor in cancer area and more drug will go to the cancer area. So nowadays we are working on dual targeting, multiple targeting. So like if you have enter a building, so there is one key for one lock and there are different kinds of locks. But if you have keys for multiple locks, your chance of entering building is more. So we are working on this theory that can we create multiple ligands on Dendrimer, like transferrin or mannose. That way we have better chance of entering brain, and that way drug will have better effect on brain. So you can see that we have done this whole study, and we have found that compared to control, this is GPM animal model histopathology. Basically, you can see that the control here, and after some time, 14 days, after GVM is developed, all the areas become cancerous and animal dies basically. But in case of our multiple targeting formulation, you can see that we were able to reduce from 100% to 84%, 0% reduction to 100% reduction. Even the Taxotere, the drug in market, is showing only 18% reduction. So basically, we were able to show, prove our theory, and we also figured out an easy way of hitting Dendrimers and creating this habitat. So, I don't want to go very deep into that. The future is like that we can create some kind of nerve design. So, what's going to be? Future would be that a system goes inside your body. It has an imaging molecule. You can diagnose what goes on the body. It may have a drug stored inside, and depending upon the requirement, it can release it. So it was able to both jobs, diagnosis and treatment at the same time, which is called theranostic. Furthermore, can be done in this area. That's what we are preparing. We are planning to prepare some sort of narrow device, a small drone inside the body. You can go inside the body and do multiple jobs. And Dendrimer architecture has the capacity to develop that kind of complex delivery system. Now I will focus on my one of the projects which I am working on. Before I start this project, explain this project to you, many of you, I just want to tell you that you need to understand that science is not supposed to be complex. Everybody doesn't need to solve breast cancer or AIDS or some Alzheimer and all those things. Around you, you can solve many simple problems. Only thing is that you should have a good idea and be honest about your work. But I'm going to show you that now, a simple idea, how I was able to bring in market. This thing can be done in any university in India and in your business. So my whole idea is that you should work on solve a problem which is solvable. You don't have to work on a fancy idea all the time. I would prefer you work on an idea which is possible in the setting you are working and try to solve that problem. You can solve a simple problem around tablets and capsules and solutions and suspensions. That will have major implication compared to doing something which is not possible doing where you are staying. So that's, I want to say that. So, before coming to academia, I was in Dendrimer company 6-7 years. I was working with Maharaj, the father of Dendrimer, many years. I still work with him. So, at the time, figured out that there is a demand in a personal care industry. There is a molecule called resveratrol. It's not an anti-aging molecule. And it's found in grapes. Grapes has resveratrol, or wine is made of grape basically. And there is a problem. This molecule, the trans resveratrol, has a solubility issue and a stability issue. And industry guys in the cosmetic or personal care industry saying, can somebody solve the problem, make us a nice formulation, what are this formulation? And what I have done, I trapped this in certain kinds of Dendrimer. I was able to increase solubility, stability, and skin permeation. Because based on my previous research, I have shown Dendrimer, and that gave me multiple patents. 1, 2, 3, 4, 5 patents are there. I published there. And also, the biggest magazine in cosmetic featured an article from me talking about use of Dendrimer for cosmetics. So I was able to increase solubility, stability of a molecule. Not only that, we have shown that we were able to increase the trans number permeation of this molecule, both in solution form, or cream and gel. So what does it mean? The current resveratrol cream can market with no guarantee that it is stable, or they are made with oil or organic solvent, which is not good for your skin or the environment. So one thing is that it's very friendly, environmental friendly, more stable, it's more permeable. What happens is that more drug will stay active, because stable, and more will cross the skin to go deep into the skin, go to the target keratinocyte cell. And then we have shown that by using the reactive oxidation assay, that compared to resveratrol alone, Dendrimer resveratrol has a better antioxidant efficacy. All anti-aging efforts come from antioxidant properties. So not only that, more is stable, more is permeating, whatsoever is going in the cell, it's more potent compared to resveratrol alone. And then we have done both gene study, all the genes of anti-aging. We studied the population, and we found that we were able to, it is doing its job. Because if resveratrol is entering there into the cell, it should be able to modulate the anti-aging genes also. So we found that it is doing the job. So a very small concentration, we were able to achieve these. And I'm explaining you the work of 8-9 years. No, because I'm a full-time faculty, so it takes time to do things here and there. Then I had to open a company and then arrange funding from Pharmaceutical place, and then we did human studies. We did clinical trial studies, and then the phase 1 study, it was found that the whole formulation in cream form is safe. And then phase 2 study, we were able to test the anti-aging efficacy of the thing compared to this. It found that it has anti-aging properties, as we were thinking about. And then last year, we were able, through my company, I was able to launch in the market. So this is the first, you can see here, this is the product available in Amazon as of now, and the first Dendrimer based product from our production market. So you can see that it's not easy. It looks easy to explain. It's not of so many barriers, regulatory barriers you have to cross and all those things. But the thing is that you can take a simple idea, bring it to market. Science has an academic input, that you can do academic science as a value, it adds the knowledge. But think about doing science, it will help people. Your science should reach people. That's what mindset some of you should come up with. For that, you have to be honest with your science. That's the reason I'm showing you all those things, to show that a simple idea can be bring into market. And now we are selling this thing in the US market to Amazon as of now. People who are buying, using it, they can feel that it has some anti-aging property, as we're thinking. And there are few Dendrimer in market, including mine in market. Mine is the first product which is in market. And also there is a book I wrote for elsewhere about pharmaceutical application of Dendrimer. If you want, it should be available elsewhere. That covers everything about Dendrimer, the basic chemistry to all kinds of applications. So with that, I'll stop. I think already I'm 30 minutes are almost over mine. So I can stop here and help you to, I'll answer any questions if you have.
Any questions from the audience, please? Nobody wants to ask anything? Okay. Thank you, sir, for sharing your valuable time and sharing your expertise on Dendrimers. So on behalf of sir, I hope Dr. Meenakshi ma'am, on the behalf of Ajay sir, please ma'am come forward to receive the mementos as well as the shawl as a token of respect on the behalf of School of Pharmaceutical Sciences, Chhatrapati Shahu Maharaj University. I invite Ajay Gupta sir. I request Dr. Nisha ma'am, please come forward. Nisha ma'am, to felicitate Dr. Ajay Chauhan sir. Please, center में आइए मैम.
On the behalf of sir, I am picking.
मैम इधर दिखाइए, इधर मैम इस तरफ.
Thank you so much, sir.
Thanks. Yeah, thanks, Dr. Gupta.
Thank you, ma'am. So, moving ahead for the inaugural ceremony, we have to wait for the chief guest. Meanwhile, I think we should proceed for the breakfast. Yes? Because we'll start the inaugural ceremony at 10:30 a.m. So please proceed for it. Or if you want to be seated, just be seated, please. And if you want to move, you can go. No problem.
नए नए पाठ्यक्रमों का सृजन, नवीन विचारों का संग्रहण तथा तकनीक व आधुनिकता के सभी सफल प्रयास विश्वविद्यालय के एकेडमिक जीवन शैली का हिस्सा बने हुए हैं। इस विश्वविद्यालय से उपाधि अर्जित करने के पश्चात यहाँ के उपाधि धारक समाज में अपनी विशिष्ट पहचान के साथ संस्थान का नाम भी रोशन कर रहे हैं। सार्वभौमिक उत्थान के लिए नित नए साझा प्रयास, जी तोड़ मेहनत एवं नवाचार के लिए प्रतिबद्ध विश्वविद्यालय को वर्तमान स्वरूप माननीय कुलपति प्रोफेसर विनय कुमार पाठक की सोच का परिणाम है, जिन्होंने अपनी दूरदृष्टि, कुशल नेतृत्व क्षमता एवं विचारशीलता से विश्वविद्यालय की ऐतिहासिक स्वरूप को तो बनाए रखा ही, साथ ही उसे आधुनिक तकनीक, कौशल के समन्वय से जोड़ दिया है। समाजोपयोगी पहल, जन जागरण के विषयों के साथ साथ विश्वविद्यालय ने जन सरोकार के अनगिनत विषयों पर जागरूकता अभियान, कार्यक्रम, संगोष्ठी, नवाचार के माध्यम से अपनी महती भूमिका निभाई है। एक किराए के भवन से अपना सफर शुरू करने वाला यह विश्वविद्यालय अपने शैशव काल से मुक्त होकर नित नए आसमान को छूने के लिए प्रतिबद्ध है। हम सभी के लिए यह गौरव का क्षण है कि ज्ञान दर्शन की इस विरासत के 57वें स्थापना दिवस पर हम इसके साक्षी बन रहे हैं। इस उत्सव के रंग में हम विश्वविद्यालय परिवार के उन सभी शिल्पकारों को प्रणाम करते हैं, जिनके अथक प्रयास से यह आज अपनी आभा बिखेर रहा है। दुनिया भर में पूरब के मैनचेस्टर के नाम से प्रसिद्ध शहर कानपुर अपनी अनूठी विरासत, संस्कृति और धरोहर के साथ वर्षों से आकर्षण का केंद्र रहा है। भारत के स्वर्णिम आभा मंडल की यह ऐसी धरती है जिसने देश की स्वाधीनता के सपनों को सींचा, ज्ञान की गंगा को राह दिखाई, क्रांति की अलख को आभा में किया और विचार की धन संपदा को अंत्योदय तक लाने का सपना साकार किया। यहाँ की ज्ञान संपदा ने दुनिया को एकात्म मानववाद की दीक्षा से संचित तो किया ही है, साथ ही युगांतकारी पत्रकारिता की आकांक्षाएं भी पल्लवित की हैं। अवतरण के ही इस प्रवाह में अगर शहर के सबसे प्रभावशाली प्रतीक को देखा जाए तो निसंदेह यहाँ का छत्रपति शाहू जी महाराज विश्वविद्यालय हम सभी की सराखों पर है। ज्ञान, प्रतिभा, विचार और दर्शन के साक्षात हस्ताक्षर इस विश्वविद्यालय के नामधारी रहे हैं। अंत्योदय के प्रणेता पंडित दीनदयाल उपाध्याय, अजात शत्रु अटल बिहारी वाजपेयी, देश के प्रथम नागरिक राष्ट्रपति महामहिम रामनाथ कोविंद का नाम इस विश्वविद्यालय की धरोहर सूची में है। यहाँ की कक्षाओं में शिक्षकों ने गणेश शंकर विद्यार्थी के प्रताप को प्रदीप्त किया है, तो साथ ही पंडित युगल किशोर शुक्ल का भारत बोध भी इसी शहर की अक्षोणी विरासत है। अपनी श्रृंखलाद उपलब्धियों के साथ कभी एक छोटे से भवन से अंकुरित हुआ यह विश्वविद्यालय आज 264 एकड़ के विशाल भूभाग पर अनेकानेक मनीषियों द्वारा शिक्षा दीक्षा के लिए जाना जाता है। उच्च शिक्षा के क्षेत्र में देश दुनिया को अपनी रोशनी से प्रकाशमान कर रहे इस विश्वविद्यालय की शुरुआत साल 1966 में हुई थी। शिक्षा जगत के मानचित्र पर आज यह विश्वविद्यालय प्रदेश के 11 जनपदों में लाखों विद्यार्थियों के भविष्य का निर्माण कर रहा है। गांव कस्बों से लेकर आसपास के शहरों तक के 600 से अधिक कॉलेज विश्वविद्यालय परिवार से संबद्ध होने के कारण इसका हिस्सा बने हुए हैं। अकेले विश्वविद्यालय परिसर में ही कला, विज्ञान, वाणिज्य, विधि, अभियांत्रिकी, चिकित्सा, समाज विज्ञान, साहित्य, पत्रकारिता, प्रबंधन, ललित कला समेत अनेक पाठ्यक्रम स्नातक एवं परस्नातक स्तर पर संचालित किए जा रहे हैं। नए नए पाठ्यक्रमों का सृजन, नवीन विचारों का संग्रहण तथा तकनीक व आधुनिकता के सभी सफल प्रयास विश्वविद्यालय के एकेडमिक जीवन शैली का हिस्सा बने हुए हैं। इस विश्वविद्यालय से उपाधि अर्जित करने के पश्चात यहाँ के उपाधि धारक समाज में अपनी विशिष्ट पहचान के साथ संस्थान का नाम भी रोशन कर रहे हैं। सार्वभौमिक उत्थान के लिए नित नए साझा प्रयास, जी तोड़ मेहनत एवं नवाचार के लिए प्रतिबद्ध विश्वविद्यालय को वर्तमान स्वरूप माननीय कुलपति प्रोफेसर विनय कुमार पाठक की सोच का परिणाम है, जिन्होंने अपनी दूरदृष्टि, कुशल नेतृत्व क्षमता एवं विचारशीलता से विश्वविद्यालय के ऐतिहासिक स्वरूप को तो बनाए रखा ही, साथ ही उसे आधुनिक तकनीक, कौशल के समन्वय से जोड़ दिया है। समाजोपयोगी पहल, जन जागरण के विषयों के साथ साथ विश्वविद्यालय ने जन सरोकार के अनगिनत विषयों पर जागरूकता अभियान, कार्यक्रम, संगोष्ठी, नवाचार के माध्यम से अपनी महती भूमिका निभाई है। एक किराए के भवन से अपना सफर शुरू करने वाला यह विश्वविद्यालय अपने शैशव काल से मुक्त होकर नित नए आसमान को छूने के लिए प्रतिबद्ध है। हम सभी के लिए यह गौरव का क्षण है कि ज्ञान दर्शन की इस विरासत के 57वें स्थापना दिवस पर हम इसकी साक्षी बन रहे हैं। इस उत्सव के रंग में हम विश्वविद्यालय परिवार के उन सभी शिल्पकारों को प्रणाम करते हैं, जिनके अथक प्रयास से यह आज अपनी आभा बिखेर रहा है। दुनिया भर में पूरब के मैनचेस्टर के नाम से प्रसिद्ध शहर कानपुर अपनी अनूठी विरासत, संस्कृति और धरोहर के साथ वर्षों से आकर्षण का केंद्र रहा है। भारत के स्वर्णिम आभा मंडल की यह ऐसी धरती है जिसने देश की स्वाधीनता के सपनों को सींचा, ज्ञान की गंगा को राह दिखाई, क्रांति की अलख को आभा में किया और विचार की धन संपदा को अंत्योदय तक लाने का सपना साकार किया। यहाँ की ज्ञान संपदा ने दुनिया को एकात्म मानववाद की दीक्षा से संचित तो किया ही है, साथ ही युगांतकारी पत्रकारिता की आकांक्षाएं भी पल्लवित की हैं। अवतरण के ही इस प्रवाह में अगर शहर के सबसे प्रभावशाली प्रतीक को देखा जाए तो निसंदेह यहाँ का छत्रपति शाहू जी महाराज विश्वविद्यालय हम सभी की सराखों पर है। ज्ञान, प्रतिभा, विचार और दर्शन के साक्षात हस्ताक्षर इस विश्वविद्यालय के नामधारी रहे हैं। अंत्योदय के प्रणेता पंडित दीनदयाल उपाध्याय, अजात शत्रु अटल बिहारी वाजपेयी, देश के प्रथम नागरिक राष्ट्रपति महामहिम रामनाथ कोविंद का नाम इस विश्वविद्यालय की धरोहर सूची में है। यहाँ की कक्षाओं में शिक्षकों ने गणेश शंकर विद्यार्थी के प्रताप को प्रदीप्त किया है, तो साथ ही पंडित युगल किशोर शुक्ल का भारत बोध भी इसी शहर की अक्षोणी विरासत है। अपनी श्रृंखलाद उपलब्धियों के साथ कभी एक छोटे से भवन से अंकुरित हुआ यह विश्वविद्यालय आज 264 एकड़ के विशाल भूभाग पर अनेक मनीषियों द्वारा शिक्षा दीक्षा के लिए जाना जाता है। उच्च शिक्षा के क्षेत्र में देश दुनिया को अपनी रोशनी से प्रकाशमान कर रहे इस विश्वविद्यालय की शुरुआत साल 1966 में हुई थी। शिक्षा जगत के मानचित्र पर आज यह विश्वविद्यालय प्रदेश के 11 जनपदों में लाखों विद्यार्थियों के भविष्य का निर्माण कर रहा है। गांव कस्बों से लेकर आसपास के शहरों तक के 600 से अधिक कॉलेज विश्वविद्यालय परिवार से संबद्ध होने के कारण इसका हिस्सा बने हुए हैं। अकेले विश्वविद्यालय परिसर में ही कला, विज्ञान, वाणिज्य, विधि, अभियांत्रिकी, चिकित्सा, समाज विज्ञान, साहित्य, पत्रकारिता, प्रबंधन, ललित कला समेत अनेक पाठ्यक्रम स्नातक एवं परस्नातक स्तर पर संचालित किए जा रहे हैं। नए पाठ्यक्रमों का सृजन, नवीन विचारों का संग्रहण तथा तकनीक व आधुनिकता के सभी सफल प्रयास विश्वविद्यालय के एकेडमिक जीवन शैली का हिस्सा बने हुए हैं। इस विश्वविद्यालय से उपाधि अर्जित करने के पश्चात यहाँ के उपाधि धारक समाज में अपनी विशिष्ट पहचान के साथ संस्थान का नाम भी रोशन कर रहे हैं। सार्वभौमिक उत्थान के लिए नित नए साझा प्रयास, जी तोड़ मेहनत एवं नवाचार के लिए प्रतिबद्ध विश्वविद्यालय को वर्तमान स्वरूप माननीय कुलपति प्रोफेसर विनय कुमार पाठक की सोच का परिणाम है, जिन्होंने अपनी दूरदृष्टि, कुशल नेतृत्व क्षमता एवं विचारशीलता से विश्वविद्यालय के ऐतिहासिक स्वरूप को तो बनाए रखा ही, साथ ही उसे आधुनिक तकनीक, कौशल के समन्वय से जोड़ दिया है। समाजोपयोगी पहल जन जागरण के विषयों के साथ साथ विश्वविद्यालय में जन सरोकार के अनगिनत विषयों पर जागरूकता अभियान कार्यक्रम संगोष्ठी नवाचार के माध्यम से अपनी महती भूमिका निभाई है। एक किराए के भवन से अपना सफर शुरू करने वाला यह विश्वविद्यालय अपने शैशव काल से मुक्त होकर नित नए आसमान को छूने के लिए प्रतिबद्ध है। हम सभी के लिए यह गौरव का क्षण है कि ज्ञान दर्शन की इस विरासत के 57वें स्थापना दिवस पर हम इसके साक्षी बन रहे हैं। इस उत्सव के रंग में हम विश्वविद्यालय परिवार के उन सभी शिल्पकारों को प्रणाम करते हैं जिनके अथक प्रयास से यह आज अपनी आभा बिखेर रहा है। दुनिया भर में पूरब के मैनचेस्टर के नाम से प्रसिद्ध शहर कानपुर
Good morning and a very warm welcome to the Honorable Vice Chancellor, sir, respected Registrar, sir, Director, Pharmacy, Dr. Shashi Kiran, ma'am. Please, sir. I would also request the organizing secretaries to join on the stage. Dr. Ajay Gupta, sir, and Dr. Anupriya, ma'am. So it's an absolute honor and privilege to stand before you, I, Dr. Shweta Singh, with my team, on the behalf of School of Pharmaceutical Sciences, Chhatrapati Shahu Ji Maharaj University, as we inaugurate this prestigious international conference on pharmaceutical frontiers, exploring future trends and technologies, pharmaceutical innovations for the new era of healthcare 2025. This event marks not just a gathering of great minds, but a convergence of ideas, innovation and inspiration. We extend our heartfelt gratitude to our esteemed guests and speakers who have taken the time to share their invaluable insight with us. Now, to mark this auspicious occasion, we proceed for the lighting of the lamp with the ceremonial symbol of wisdom, knowledge and enlightenment. So, to do the honor, I request the Honorable Vice Chancellor, sir, respected Registrar, sir, Dr. Nisha, ma'am, Director, ma'am, Dr. Ajay Kumar Gupta, sir, Dr. Anupriya Kapoor, elegant guests and distinguished persons, please proceed for the lighting of the lamp. I would like to give an introduction about our university, department as well as about the event. So we would love to make our audience aware of the glorious history of CSJMU, that is Chhatrapati Shahu Ji Maharaj University, Kanpur, established in the year 1996, is a leading state government university in Uttar Pradesh. It is accredited with NAC A++, acquiring 263rd position in the international QS South Asia rankings. CSJMU offers an impressive range of 400 undergraduate courses along with diverse postgraduate and doctoral programs, all within its expansive 264-acre campus with 17 specialized departments, including the Department of Education, the Department of Computer Science, and the School of Languages. The university stands as a hub of multidisciplinary learning. CSJMU is dedicated to implementing the new National Education Policy NEP 2020, striving to expand the horizons of knowledge while being enriched by a profound legacy of literature, art and culture. Committed to academic excellence, the university actively fosters an educational ecosystem that aligns with the spirit of sustainable development goals and the vision of Atmanirbhar Bharat, empowering students for a self-reliant future. The School of Pharmaceutical Sciences is recognized in the NIRF ranking, which is dedicated to advancement in the field of pharmaceutical education and research. Established with a vision to cultivate skilled professionals, the school offers all courses like D Pharma, B Pharm, M Pharm, as well as PhD programs that equip students with a strong foundation in pharmaceutical sciences. The School of Pharmaceutical Sciences emphasizes interdisciplinary collaboration, fostering partnerships with industry and research institutions. The International Conference on Pharmaceutical Frontiers, Exploring Future Trends and Technologies, Pharmaceutical Innovations for New Era of Healthcare, organized by School of Pharmaceutical Sciences, Chhatrapati Shahu Maharaj University, offers a unique opportunity to connect with pioneers, researchers, as well as policy makers, fostering collaborations and knowledge sharing. The PFEFT 2025 features keynote speeches from eminent academicians that cover diverse research areas, including innovations in drug discovery, AI in computer-aided drug designing and docking, medical microbiology, innovation and future trends in pharmaceutical formulations, herbal drug technology, medical biochemistry, innovation in education, science, role of personalized medicine, current research in pharmacology and drug discovery. Recent trends in biological sciences, innovation in naturopathy, advancement in community pharmacy, entrepreneurship skills development, traditional folklore medicine. In today's conference, we have a series of exciting events lined up, including our 4 insightful lectures which will be delivered by our eminent speakers in the order of Dr. Abhay Singh Chauhan, that has already been held because of the time zone difference, Dr. Kamla Pathak Madam, Dr. Alok Mukherjee Sir and Dr. Monica Arora Madam, respectively, followed by a poster and oral presentations. So we look forward to an enriching and engaging experience for all. Thank you. Now, we'll move ahead for launching of our environment-friendly e-souvenir. So, to do the honor, I request the Honorable Vice Chancellor Sir, respected Registrar Sir, Convener Dr. Shashi Kiran Ma'am, Co-Convener Dr. Nisha Ma'am, and of course, Nisha Ma'am is the one who took all the painless effort for the launching of this souvenir. So I request all of you to please, technical team, please do the honor. Yes. Nisha Ma'am, please come forward. First page, please. Big round of applause, all of you. Thank you, sir. Thank you, ma'am. Now, moving ahead, I would like to invite respected Registrar Sir to please. Okay, sir, I'll call direct. I request Director Pharmacy Dr. Shashi Kiran Ma'am to please address the gathering.
Good morning to all. मंच में आसीन हमारे ऑनरेबल वीसी सर, रेस्पेक्टेड रजिस्ट्रार सर, हमारे ऑर्गेनाइजिंग कमेटी के डॉक्टर अजय गुप्ता सर एंड अनुप्रिया कपूर मैडम, और हमारे जो आज के चीफ गेस्ट हैं डॉक्टर प्रोफेसर कमला पाठक मैडम, डॉक्टर आलोक मुखर्जी सर, सीएस भार्गव सर, रेस्पेक्टेड मोनिका मैडम और सारे मेरे पार्टिसिपेंट्स, मैं सभी का यहाँ पे स्वागत करती हूँ। स्कूल ऑफ फार्मास्यूटिकल साइंस की तरफ से आप सभी का आभार है, आप प्लीज यहाँ पे आए हैं। दिस इज वेरी ऑनर टू अस। और मैं एक्सपेक्ट करती हूँ कि जो हमारा एक दिवसीय जो अंतर्राष्ट्रीय सेमिनार जो हम आयोजित कर रहे हैं वो आप सभी के लिए बहुत बेनिफिशियरी होगा। सेमिनार का मतलब यही होता है कि एक्सचेंज ऑफ इंफॉर्मेशन, एक्सचेंज ऑफ नॉलेज और नए नए जो आयाम स्थापित हुए हैं फार्मेसी विधा में उसको हम लोग एक्सेप्ट करें, सुने, देखें, समझें और उसको इम्प्लीमेंट करें। तो इन्हीं सभी विचारों के साथ मैं अब श्वेता मैडम को माइक हैंड ओवर करती हूँ। मैडम आगे प्रोसीड करें।
थैंक यू मैम। Now I request Dr. Ajay Gupta sir, organizing secretary of this event, to say few words and address the gathering. Please sir.
धन्यवाद। सर्वप्रथम मैं हमारे यशस्वी कुलपति, विश्वविख्यात कुलपति जी श्री विनय पाठक जी का हार्दिक स्वागत अभिनंदन करता हूं। और आज ये जो हमारी महफिल सितारों से सजी है, इन सितारों में जो रोशनी है वो भी AKTU के पूर्व कुलपति और हमारे वर्तमान कुलपति प्रोफेसर विनय पाठक जी की वजह से ही है। क्योंकि AKTU की हालत उस समय क्या थी जब सर ने ज्वाइन किया था, और उसके बाद आज हमारे कानपुर के जितने भी इंस्टीट्यूट हैं AKTU से संबंधित, और हम में से ज्यादातर सभी लोग डायरेक्टली इनडायरेक्टली AKTU से संबंधित रहे हैं और अभी भी हैं। तो आज हमारा कानपुर विश्व के मंडल पर जो प्रकाशमान है वो हमारे माननीय कुलपति जी के विजन की वजह से ही है। और हमारे विश्वविद्यालय के जो कंगूरे पे जितने भी सितारे लगे हुए हैं, चाहे वो NAC A++ की रैंकिंग हो, इंटरनेशनल रैंकिंग हो, NIRF हो, वो हमारे कुलपति महोदय जी के मार्गदर्शन और उनकी दूरदर्शिता की वजह से ही संभव हो पाया है। आज की ये कॉन्फ्रेंस भी आपकी प्रेरणा से, आपके संरक्षण में हम आयोजित कर रहे हैं। और हम बीइंग द ऑर्गेनाइजिंग सेक्रेटरी ऑफ दिस कॉन्फ्रेंस और पूरे विश्वविद्यालय परिवार की तरफ से आप सभी का स्वागत करते हैं, अभिनंदन करते हैं। धन्यवाद।
Now I request Dr. Anupriya Kapoor, organizing secretary of this event, to say a few words. Please, ma'am.
Thank you, first of all, Shweta ma'am, for giving me this dais to speak something. First of all, I need to thank the Lord Almighty कि उन्होंने मुझे आप सबके सामने खड़ा करने की हिम्मत दी है। Then the first and the foremost thanks is to the honorable Vice Chancellor sir, because उन्होंने शायद मुझ में ये पोटेंशियल देखा और उन्होंने ही कहा था कि अनुप्रिया तुमको भी ऑर्गेनाइजिंग सेक्रेटरी बनना है। रजिस्ट्रार सर एंड गुप्ता सर एंड शशि किरण मैम, उन्होंने मुझे हर लेवल पे हेल्प किया। आप सब डिग्निटरीज, एमिनेंट स्पीकर्स, आप सब लोगों ने विद अ वेरी शॉर्ट फ्रैक्शन ऑफ टाइम हमारा जो एक रिक्वेस्ट था टू बी अ पार्ट ऑफ दिस इवेंट एक्सेप्ट किया। इट्स रियली वंडरफुल। मोरओवर आज यहाँ पे नियर अबाउट 172 ओरल एंड पेपर प्रेजेंटेशंस होने वाले हैं एंड इट इज एक्सपेक्टेड कि नियर अबाउट 20 to 25 पेपर्स आगे भी जाएंगे। And each and every member of this organizing team, you all have helped us a lot. Thank you VC sir and thank you Registrar sir.
Thank you very much. Now I request respected Registrar sir to say a few words. Please sir.
Chhatrapati. Good morning to all of you. On the dais, Honorable Vice Chancellor Professor Vinay Kumar Pathak ji, Director of this school Dr. Shashi Kiran Mishra ji, Dr. Ajay Gupta ji, Secretary, और Anupriya ji. We are here to inaugurate the international conference Pharmaceutical Frontiers, exploring future trends and technologies, pharmaceutical innovations for new era of healthcare. Here, the department has invited the expert Dr. Abhay Singh Chauhan ji from US, Dr. Kamla Pathak ji, I think Itawa from Shahu, Dr. Alok Mukherjee, Prayagraj, Dr. Monica Arora ji, Maldives. So, on behalf of the university, we are welcome to all of you here in this gathering. all the faculty members of this school, and dear students, research scholars and media persons. सबसे पहले तो आज महिला दिवस है, सभी लोगों को बहुत बहुत हार्दिक शुभकामनाएं और बहुत बहुत बधाई कि इस अवसर पे आप लोग इस इंटरनेशनल कॉन्फ्रेंस को आयोजित कर रहे हैं। तो डेफिनेटली जो फार्मास्यूटिकल का फील्ड है निश्चित रूप से बहुत ही डिमांड में है आजकल। हम लोग टेक्नोलॉजिकल का यूज कर रहे हैं, टेक्निक का इसमें कैसे यूज करें, कैसे इसको बढ़ाएं, स्टूडेंट्स को जो अपने हैं उनको कैसे एक्सचेंज आइडियाज करें इस कॉन्फ्रेंस के थ्रू, और कैसे इसको नई न्यू स्किल्स के बारे में हम लोग बताएं। अभी रिसेंटली कल या परसों एक बीसी सर ने एक एलएनटी की टीम बुलाई थी, उसने भी बहुत सारे स्किल बेस्ड कोर्सेज डिजाइन किए हुए हैं। तो उसको हम लोग कैसे बच्चों तक ले जाएं, उसको बच्चे कैसा फीडबैक दें, उनको लर्न करें, हाउ टू लर्न द न्यू थिंग्स, क्योंकि आजकल बहुत रैपिडली टेक्नोलॉजी चेंज हो रही है। रिसेंटली जैसे चैट जीपीटी की बात करते हैं या डीप सीक की बात कर रहे हैं, तो बहुत स्पीड से हम लोगों को बहुत ही तेजी से काम करना है इस फील्ड में, क्योंकि फार्मास्यूटिकल सेक्टर है वो भी रैपिडली चेंज हो रहा है। तो उसमें कैसे लैब में क्या क्या आपके एफर्ट्स हैं, उसमें कैसे डेयरी कर रहे हैं या कैसे हम लोग सॉफ्टवेयर के थ्रू उसको सिमुलेशन कर रहे हैं, तो इन सब के सारे बच्चों को, स्टूडेंट्स को वी हैव टू फोकस ऑन दिस। और टेक्नोलॉजिकल चाहे आर्टिफिशियल इंटेलिजेंस की बात करें या मशीन लर्निंग की बात करें, तो हाउ टू डेवलप सच काइंड्स ऑफ इन दिस फील्ड, तो इन पे हम लोगों को काम करना है। कॉन्फ्रेंस के करीब अभी डायरेक्टर मैडम ने बताया कि मोर देन 150 पेपर्स हैं, कुछ 20-25 और पेपर हैं। तो कॉन्फ्रेंस सक्सेस हो रही है, लेकिन अभी हमको इसमें चल के और अच्छे मेहनत से करना है, काम करना है। तो डिपार्टमेंट के सभी फैकल्टीज को, डायरेक्टर को, सेक्रेटरीज को बहुत बहुत शुभकामनाएं और बधाई कि आप लोग ऐसे ही अच्छे काम करें और स्टूडेंट्स के फेवर में काम करें, उनके हित में काम करें, जिससे कि वो मार्केट में जाके अपना एक प्लेस पा सकें। थैंक यू सर।
Thank you, sir, for your insight. Now, before I call upon the Honorable Vice Chancellor, sir, to enlighten with his valuable thoughts and experience, we would like to pay tribute to our Honorable Vice Chancellor, sir, because without his guidance and inspiration, it wouldn't be possible to execute this event. So, with your permission, sir has emphasized his innovation.
Thank you.
So, thank you.
Thank you very much for inviting me in this international conference. I don't want to talk because I am not a subject expert in the pharmaceutical sciences and pharmaceutical engineering. First of all, I want to really appreciate the speakers who have come, all the 4 speakers, the eminent speakers who have come and joined us in this journey. I really wanted to congratulate the organizing secretary, Mr. Gupta, the Anupriya ji and Anuji and Shashi ji for holding such a nice seminar. I think this seminar and the workshop has got 172 oral presentations, 432 participations, and 20 to 25 papers are going to be published in the reputed journal. So congratulations to all. I think I will start from a quote which Nokia CEO gave, and I used to talk to about these things already then. When Nokia giant and Nokia telephones and Nokia mobile was over and it was brought to the Microsoft. Microsoft was buying that whole product. Then the Nokia CEO told that we were strong, we did nothing wrong, but we still failed. I think that speaks louds and volumes that even we are right, even we are strong, but if we are not competitive in this world with our competitors, I think we will be finished. This is what that Nokia CEO was trying to say. Similar are with the institutes, similar are with the technologies, what Anil was telling about ChatGPT, DeepSeek and all other things. In the pharmaceutical industry, when Corona came and whole world thought that why not to make their own pharmacies and own pharmaceutical drugs, then it came to mind because India was a giant sometime before when including this pharmaceutical industry. But then we thought that the molecule or the raw material we have to buy, we are not very competitive in the world to buy those raw products. And that's how we lost to China in those kind of things. We were thinking that we can make, but in this competitive world, whether we can make those things or not, this is the question. When we are hearing about these reverse tariffs and all other things, then it looks to me that the whole world is governed by two important things. One is new design and discovery. When you have got patents, with those patents you have got wealth. The other, you can produce in a competitive prices. And that is a kind of example where most design and patents are with the US and most of the production is with the China. So if you look around, then where is our country placed? Where are our researchers placed? I think it's a high time when we should look into the universities, into the things. One of my students today in the morning was telling me he was in US. He said the only thing that Taiwan has, it's not a country, but then it has got the whole world of making semiconductors. It has got the fab lab of semiconductors. And that's why whole world, including the China, is behind Taiwan. And that's why the Taiwan is a developed country and they are a strategic country because they produce semiconductors. So then where we are placed? So we have got good institutions, we have got very good researchers, we have got good labs, but do we have so much patents where we can get them? Do we have some papers which need to be translational research? Whether we have got papers or we have got product, or we have got product from papers. These are very, very important points at the juncture which we have to really think. I was talking to Ajay that if B Pharma and M Pharma or Diploma of Pharma goes, students don't see that they want to do this course, what are the other options we have so that we can boost the pharmaceutical industries? And this is the right time. We do publish researchers, we do publish patents, but we then don't have wealth out of those patents and papers. I think this is a high time for all of our researchers and youngsters, because youngsters always believe that if I do my B Pharma course, what next? Where I will placed? Where is my job? Can I do with the industry? Can I do these things? And then this is a job of a teacher or a facilitator or researcher or research scientist to give a paved path to those things. And I think it's a high time for our country to think of productions, to think of new designs and think of new patents. When AI has taken over the whole world, when AI is doing disruption, there are a lot of opportunities with our pharmaceutical people to make new designs, to make new precision medicines, to make new medicines which are target oriented. And that's how our all pharmaceutical people or all our pharmaceutical scientists have to learn technologies. If they don't know technologies, they will not be able to cope up with those people, those scientists who knows technology, who can do research overnight, who can take lakhs and crores of data points and publish or something in a new kind of thing. So we are in a great competition. The whole world is in a great competition. It may be possible that we are sitting today in this building. It may be possible that if we don't compete properly, this department may not exist after 10 years, 20 years, 30 years, like we are seeing a lot of things in Kanpur. So to make it competitive, to make it that it lasts for a long time, you should be on toes. Heavy researchers, good researchers, researchers by M-Pharma students, researchers by undergraduates, patent filing and product filing. And it should be Department of Translational Research, where we turn papers into a product, where we take consultancies, where we are aggressive in doing new research. I think these are the key notes and key things and key points which our country really need at this juncture. We have got youth dividend, We are a country of youth. We are not like Europe, where only old people are left. But then we have to see what our competitors are doing, especially the South Indian countries, South Asian countries like China, Hong Kong, Singapore, and all other things. Seeing what European countries are doing, what US and Canada is doing, how their researchers are working. And this is the high time. This can be only achieved by departments, only achieved by academics. As a felicitator, as a vice chancellor, and as a registrar, we are actually want to say that if you have got any new ideas, if you have got any new design, then as a felicitator, as a vice chancellor, we will see to it last time that you don't have any dearth of money for doing research, you don't have any dearth of doing equipment, all those things, all those facilities are our responsibility. But then you have to come with ideas, you have to come with products, you have to come. Thank you very much and best of luck for this great seminar.
Thank you, sir, for your blessings and aspirations. As we proceed forward, so now it's time to felicitate our chief guest. So to do the honor, I would request Dr. Shashi Kiran ma'am, Director Pharmacy, Dr. Ajay Gupta sir, Organizing Secretary, Dr. Anupriya Kapoor ma'am, Dr. Nisha ma'am, to felicitate the Honorable Vice Chancellor sir. Please sir, please ma'am, come on the stage. And respected Registrar sir. Thank you, sir, for accepting as a token of gratitude and respect. Thank you, sir. Thank you, ma'am. Please have a seat. So now so this is a transition phase. We have to just prepare the stage for the lectures. A few as a Dean and Professor, Faculty of Pharmacy, Uttar Pradesh University of Medical Sciences, Etawah, India. She is a member of various professional bodies, actively engaged in research and oral control, modulated, targeted and topical drug delivery systems. She has over 300 publications in reputed journals, international as well as national, 3 patents. She has in her record 3 patents, 4 books, 50 book chapters in international books and more than 180 abstracts of the papers presented in various scientific forums are to her credit. She has been credited as a reviewer of national and international journals of reputed in the field of pharmaceutics, having a H index of 44 Scopus and 54 Google Scholar, and has served as a member of editorial advisory board of AAPS Pharma Science Tech. She has handled various research projects and has guided PhDs and 120 plus postgraduate theses. Award bestowed upon her included Dr. R.L. Nikor Award 2012 for Best Research Paper in Pharmaceutics, Mutan Devi Dandiya Prize for Best Research Publication in Pharmacy 2012 and 13, Eminent Teacher Award in 2012, Nirali's Inspiring Pharma Personality Award in 2019, Research Excellence Award in SPER 2019, and for various co-authored poster presentations and national pharmacy conferences held in India. She is also associated to Indian pharmaceutical industry in the capacity of scientific consultant. Credited as a reviewer of scientific projects, Knowledge Foundation, Sweden, Israel Science Foundation, and she is being an invited member of American Nano Society. She has participated in various national and international seminars, conferences in the various capacities as organizer, invited speaker and chairperson of scientific sessions. She has been enlisted in top 2% Indian scientists in the subsequent pharmacology and pharmacy in a list released by Stanford University in 2020, 21, 22, 23, 24. Thank you, ma'am, and my apology if I have been mistaken somewhere. So I welcome ma'am to deliver her lecture. Please give a round of applause, please, everyone.
Very good morning to everyone. Good morning, the Good morning. So, first of all, I extend my warm greetings to the organizers of the conference and also to the director of the Pharmaceutical School, Dr. Shahu, who was my student way back in 1999, starting from then. So it is like always homecoming here because I know most of the faces here. And it's a good thing that the first time we have organized an international conference with a very update theme of advances in pharmaceutical sciences, and I have chosen a topic to speak on 3D printing of drug delivery systems. Now in pharmacy we all know what is a drug delivery system, but I want to talk about the 3D printing of the drug delivery systems. And as our Honorable Vice Chancellor was telling, that we have to look to the future. कि ये डिपार्टमेंट कहीं बंद ना हो जाए इस टाइप से ना कि फार्मेसी का डिपार्टमेंट बंद ना हो जाए, बट अगर हम नई नॉलेज इंटीग्रेट करते रहेंगे तो ये क्वेश्चन नहीं आएगा. तो 3D प्रिंटिंग इज वन ऑफ द आंसर्स फॉर सस्टेनेबिलिटी. So what is 3D printing? It is basically layer by layer production of 3D objects from digital designs, and it is an area which is actually confluence of chemistry, optics, robotics, research. And it was established more than 35 years ago and is quickly adopted by automobile industry, aerospace, consumer goods. But in pharmaceutical manufacturing, it came into existence when the FDA approved a 3D printed drug product in August 2015. And research interest in 3D printed product has been growing since then. So what is 3D printing? It is basically we want a tablet which is printed. It is a printed tablet and the printing is a 3D printing. So there has to be a process. पहले उसको डिजाइन करना है on computers through software और फिर उसको प्रिंट करना है. प्रिंट करने के लिए नॉर्मल प्रिंटर्स ही यूज होते हैं जो इंकजेट लेजर प्रिंटर ये. So it may sound very strange, but I'll let you explain what is happening. First is you need a 3D CAD file which is converted to layer by layer .stl file. Then comes the AM process where the printing takes place layer by layer to get the final object. So this is the theme of the 3D printing, which is also called as additive manufacturing. So first thing is you design. The intended product design is digitally created by computer aided design software or in 2 dimensions. So either you can prepare 3D or 2D. Then convert the design to a machine readable format, which is the STL format. तो जो कंप्यूटर पे डिजाइन है उसको मशीन रीड कर सके. 3D प्रिंटिंग एक 3D प्रिंटर एक मशीन है, तो मशीन रीड करे ऐसे डिजाइन में उसको कन्वर्ट करना है. दैट इज STL फाइल. And then 3D printing programs, what they do is they slice the entire object. जो 3D डिजाइन है उसको लेयर बाय लेयर स्लाइसिंग करनी है, क्योंकि प्रिंटिंग भी लेयर बाय लेयर ही होगी. And these instructions are transferred to the printer. Now you need the raw material which is to be created into a dosage form. For that you need granules, filaments or binder solutions to facilitate the printing process. जो आपको granulation में चाहिए, tablet granulation में वही चीजें यहाँ चाहिए. Then the raw materials are added and solidified in an automatic layer by layer manner to produce the desired product. Then after printing, the products may require drying, sintering, polishing or other post-processing steps. Like in tablets also, after manufacturing we polish them. उसी तरह से करना है. And if there is any material which is unprinted, it may be harvested and recycled. So there are various methods of 3D printing. Some of them are binder deposition, material jetting, fused filament fabrication, hot melt extrusion, powder bed fusion, photopolymerization, pen-based 3D printing. Now this is one thing which you can order on Amazon also. You cannot print the tablet, but there are 3D printing pens available on Amazon. And use of 3D printed molds. So all these 3D printing methods, they basically involve 2 basic steps. One is that you design the object via computer software. That is AutoCAD, 3D Slash, SketchUp, Fusion, 360, SolidWorks. Some of the softwares are free online. And then you have to slice it. Slicing the form design through slicer software like KIS Slicer, Slick 3R, Octoprint. इससे slicing होगी computer पर ही. And in addition to this, it basically what it is doing, it is converting into an STL file. And it also sets the printing parameters. की speed क्या होनी चाहिए printing की, layer की printing कैसे होनी चाहिए, and what should be the space between the layers. Everything is controlled by the softwares. Then the second step is the object deposition or formation via 3D printer. So जो हमने यहाँ create किया है STL file, that is called as the G code. अब ये G code में जब तक convert नहीं होगा तब तक printer उसको read नहीं कर सकता. So the G code is then uploaded to the 3D printer, and with the print command, the printer will run and form the object of the desired size and shape. So the object deposition process through 3D printer varies based on the type of 3D printer used. So as I told you, the printers can be inkjet based system, extrusion based system, and laser based system. It's you are very familiar with the first one and the last one. Now there are more methods of 3D printing, but they are not used for drug product manufacturing. Those include directed energy deposition, laminated object manufacturing, electrospinning, extrusion plus electrospinning plus molding, and voxel printing. So these are used in other areas, other industries like automobile industry or home goods industry, consumer products, but drug manufacturing में नहीं use हो रहे हैं. So 3D printing, we come down to 3D printing in drug delivery. It can be used for both hydrophilic and lipophilic drugs. It can be used for creating drug delivery systems of BCS class 2 and class 4 drugs, which are very troublesome for normal formulation. And we can use the 3D printing to improve solubility and bioavailability profiles. And the interesting and the beauty of 3D printing is that we can create unique, novel and specific geometries tuned with the tailored drug release characters. जो जिस तरह की characters drug release चाहते हैं आप वैसी मिल जाएगी, and the profiles can be modified. So these are the dosage forms which can be 3D printed, like IR tablets, SR tablets, modified release tablets, IR films, transdermal implants, transdermal films, pulsatile release capsular systems, and CR implants. So these are some examples which I have given that these can be produced by 3D printing. So before I go to the commercial aspect, I would like to exemplify some research areas. So this is one research example in which a 3D product was created for the anti-diabetic drugs. The drugs are metformin and glimepiride. They were used as model drugs. And the rationale was that the co-administration of these APIs pose major challenge. What are the challenge? First is they have different dosage regimen. For example, glimepiride is given only once a day, whereas metformin 2 to 3 times daily. Then the daily dosing also differs. For example, glimepiride is 1 to 4 mg, whereas metformin can range from 500 to 1000 mg. Again, the absorption site is also different. For glimepiride, it is in gastric environment, and metformin in small intestine. So they have variable profiles for absorption, dosing, and the dosage regimen. And if we want to administer them simultaneously, it is a problem, and this can be solved by a bilayer design consisting of metformin-loaded Utrajet RL layer for prolonged release and a glimepiride-loaded PBA layer for immediate release. So this diagram shows what has been done. We have created the design, and this is the printer, 3D printer, which has printed the bilayer tablet. And what we have done is metformin is mixed with Utrajet RL and glimepiride with PPA, and both simultaneously are added to the 3D printer to get the bilayer tablet like this. तो एक ही printer से एक tablet print हो रही है, आपको दो machines नहीं चाहिए. And there are 2 layers, one for immediate release and the other one for sustained release. Now once the tablet has been produced, we need to do the quality control. कि जो हमने design कंप्यूटर में डाला था वही हमें मिला कि नहीं. And for that we use microfocus computed imaging based actual nominal comparison analysis. So this tells us कि जो design हमने create किया था कंप्यूटर पे वही design print हुआ. Then XRD will tell us what is the state of drugs, whether it has changed from crystalline to amorphous, and then dissolution test to check the release profile. So this is how we do the 3D printing, and this is one research example. I have given the reference also. Then there is another one which is a novel 3D printed pressure controlled drug delivery system. Again, the concept is that we want to have a gastro-retentive system, and this system has been 3D printed in capsular form. And the release is controlled, that it releases, delayed release of the drug is observed for colon targeting. Now, why do we need 3D printing? So there are 3 drivers which motivate the 3D printed products. One is product complexity, next is personalization, and on-demand manufacturing. So I'll try to explain these so that you get into a picture in your mind that why 3D printing is justified. First is increased product complexity. See, pharmaceutical dosage forms, since we have studied, they have evolved from simple to very complex, and we have harvested botanicals to ointments, powders, lotions, compressed tablets, and these tablets were first produced in 1878. Since then, the polymer science, as it entered into 20th century, we got extended release, delayed release tablets, transdermal systems, and long-acting implants. Then came the era of 3D printing of pharmaceuticals, which was first reported around 1996, which is basically digital control over the arrangement of matter. Earlier, it was only the mechanical control of the matter. And it is a step change in dosage form evolution that will produce striking changes in immediate release modified release and combination drug products. So what are the highlights of 3D printed products? See, the product structure can affect the drug release. We know that. So by 3D printing, we can produce complex 3D structures, which we cannot produce by conventional methods. Then in this series, FDA has approved Spiritam. It has been marketed also as it is a tablet porous structure produced by 3D printing process that binds powders without compression. And this structure allows tablets with up to 1000 mg of the drug to disintegrate within seconds. So this is the beauty of 3D products. Then again, we need to understand that disintegration is a quality attribute which is affected by structure. So if we can produce complex 3D printed shapes, we have speedier dissolution than traditional immediate release products. And we can also use strategies for increasing the dissolution rates, which can be printing high surface area shapes. So we can produce, by use of 3D printing, we can produce high surface area which can be easily dissolute. And we can also print amorphous dispersion by hot melt extrusion. Now 3D printing may also create new manufacturing options for potent APIs. Generally when we produce formulations from potent substances, there is always the risk of hazard of the dust particles with the potent APIs. So 3D printing is a solution to that. It will reduce the hazard. Then printing of extremely low dose products, like as low as 3 nanogram of API with 10% relative standard deviation. We cannot achieve this by conventional process. 3 nanogram की तो tablet नहीं बना सकते. Then again, fluid bed granulation can produce 1 microgram tablets with 5% relative standard deviation. इसकी IP में 20% तक variation permitted है. But 3D printing से you can be very close, and no conventional process can produce the tablets with such precision. 3D printing can also be used to produce drug devices and drug biological combination products. Now, these are very complex products, and for producing them, digital control is very necessary. So we can produce drug devices like wirelessly triggered capsules, magnetically controlled microtransporters, antibiotic loaded catheters, and also drug biologic combination products like drug eluting scaffolds for pharmacologically controlled tissue engineering. So try to understand the area where we can work. Next driving force for 3D printing is personalization. What is personalization? Means we are giving personalized medicine based on the biomarkers. For example, Herceptin is used to treat HER2 overexpressing breast cancer. And because we know that HER2 is being expressed in breast cancer, we can design a 3D printed dosage form. So, and according to the patient need. So 3D printing facilitates personalization because we can have personalized dose and personalized dosage form. So 3D printing can make multiple small individual batches which are economically feasible. And it enables personalized doses, personalized implants, and personalized products. Ensures accurate dosing in children and permits personalized dosing of highly potent drugs. So Precision Medicine Initiative was taken up in 2015 in USA, which was supported by then President Obama. The vision was to increase the research efforts to take into consideration the differences between the people in the matter of gene, metabolism and lifestyle. Currently क्या है? One size fits all. हम कोई भी टैबलेट है पूरी दुनिया में एक ही तरह की टैबलेट घूम रही 500 mg पैरासिटामोल. Nobody is bothering कि किसको क्या चाहिए. So the current is one size fits all is non-compatible with the solutions to implementing precision medicine. So 3D printing can produce small batches with precise doses and release profiles, and it is a promising solution for implementation of precision medicine. So I have shown some examples of precision medicine where we can combine all of the patient's medication into single daily use. So this one shows that we can combine ramipril, atenolol, pravastatin and aminosalicylic acid and hydrochlorothiazide, one single tablet, in various compartments, each having different release profiles. Likewise, we can create a polypill जिसमें कि आप देख रहे हैं कई layers हैं. जितनी layers हैं उतनी drugs हैं. 1, 2, 3, 4, 5, 6, 7. 7 drugs combined together as one pill. This is possible only by 3D printing. Likewise, here also you can see different layers. 2 drugs in various variable release profiles have been combined by 3D printing. Again, as I told you, we can create personalized medicines for children also, and we can create medical devices like tracheal splints, bone grafts, and personalization will, what is it going to help? It will improve patient compliance. And to demonstrate this, the researchers at University College London printed animal-shaped drug products for children. So these are not toys, these are actually medications. And they have been printed specially for children because बच्चों को दवाइयाँ बहुत मुश्किल से दे जाती हैं, तो उनको फ्रेंडली वे से दे रहे हैं in form of toys, shaped as little animals. The next slide, it shows what can be created by 3D printing. We can have drug-loaded implants. You can see here. These all pictures are telling us even the bone graft, printed bone graft है, titanium nanotubes है for delivery when there is a fracture in the hip bone. So all the possibilities are possible. You know, it is like sky is the limit with 3D printing. Likewise, we have personalized medicine for children, as I told you earlier, and there is a firm called Fabrics who is producing such formulations, and it is producing a 3D printed product for a disorder called maple syrup urine disease. And it has printed its chewable printlet tablets with braille, which is not possible with conventional. तो अंधे लोग भी देख सकते हैं, मतलब समझ सकते हैं कि क्या टैबलेट हम ले रहे हैं. ब्रेल प्रिंटिंग हो रही है उस पे, एंड लाइक मून पैटर्न्स ऑन द सरफेस टू एड मेडिसिन टेकिंग फॉर पेशेंट विद विजुअल इम्पैयरमेंट. So the company has launched its 3D Dendrimer 3D printer in April 2020, which is specifically designed for manufacture of personalized drug delivery devices. Third driving force is on-demand manufacturing. So 3D printing could be beneficial for public health because we can directly print anything on the patient. And this has been applied to create tissues, engineering scaffolds and wound healing gels on demand. जब चाहिए तभी प्रिंट कर लीजिए. ऐसा नहीं कि बना के रखा है, स्टॉक मार्केट में है, प्रिपेयर कर रहे हैं, पूरा डिस्ट्रीब्यूशन चैनल. You don't require that. So printing in time and in other resource constrained settings. We can do in disaster areas, 3D printing, emergency rooms, operating rooms, ambulances, intensive care units, and in military operations. Kahin bhi, in the place of emergency, we can use 3D printer. Then again in formulation development in industry, again we face the crisis कि बहुत जल्दी product चाहिए और कम cost में बन जाए, तो 3D printing is one answer. Even printing low stability drugs for immediate consumption, that can also be done. And this began as early as 2011. This concept is applied to 3D printing in 2014. For example, 3D printing of nitroglycerin tablets. So this slide is an interesting slide which is showing direct 3D printing on the skin. Burn है, उसको कंप्यूटर पे डिजाइन आ गया है, and then it can be whatever you have to treat it. Skin grafting कर सकते हैं, tissue engineering कर सकते हैं. So there is no need of OT and all. That 3D printing से skin print हो जाएगी. Now coming to the commercial angles, it is not a theory, it is not a concept. There are pharmaceuticals which are producing, like Appreciate Pharmaceutical. It has received FDA approval for its 3D printed product, Spiritam, which I told earlier, in 2015, which is the first 3D printed product for seizures. And it has been approved for commercial manufacturing operations. Then it has, the company has announced a long-term collaboration with R&D firm Backlight to expand its capabilities with 3D printing. And they wanted to take it to the commercial scale, and they have also partnered with a Cambridge-based Cycle Pharmaceuticals. Then this company, Triastex, it is a Chinese-based pharmaceutical concern which has 3D printed products. They are called as T19, T20 and T21. They are through various phases like IND se cross karke, they are come to NDA. This is in IND clearance by FDA, and third one also is in the IND clearance. So 3D printed T19, it has got its IND approval from US FDA in 2021. It is basically a chronotherapeutic drug delivery system for treatment of rheumatoid arthritis. The technique used is melt extrusion deposition technology platform, and it's a complex design with internal geometry which allows the control of onset of time, duration and overall release profile. बहुत complex design भी बन सकते हैं. And it has plans to apply IND approval in China, Japan and Europe. And the NDA application has been filed in US FDA in 2023. बहुत पुरानी बात नहीं है. Then we have 3D printed T20. It provides one daily dosing with efficacy same as the currently marketed product, which is to be given 2 times. तो इसमें एक डोज ही देनी है. Treatment of cardiovascular and clotting disorders. Digital formulation development process used is 3D FED. And again, the internal geometry has been created using melt extrusion deposition technology to achieve desired release profile and pharmacokinetics. And it has been at near the top of the global sale for the past years. This is the third product of Cryostech, which is T21. It is again using melt extrusion deposition technique. And if you see the figure, there are various layers: enteric layer, delay layer, and drug coat. एक ही मशीन से प्रिंट हो गए सारे. ठीक है, बहुत सारी मशीन नहीं चाहिए कि एक कोर बनाएगा, फिर उसको डिले लेयर पे डालना है, फिर उसको कोटिंग करनी है. एंड इट हैज बीन डिजाइन टू ट्रीट सीवियर अल्सरेटिव कोलाइटिस. एंड जो भी ट्रायल्स हुए हैं स्पेशली विद इमेजिंग, तो इसमें क्या किया है कि इसमें रेडियो लेबल कर दिया इसको, सो दैट वी कैन टेक एन एक्सरे रेडियोग्राफी स्टडीज करनी है. और उस प्रोडक्ट को कहते हैं T23, जिसमें कि रेडियोएक्टिव एलिमेंट ऐड किए. Apart from these examples, elsewhere Merck has also announced plans to work with EOS Group to produce 3D printed tablets for clinical trials, then later for commercial manufacturing. Research papers, you will find abundant research papers on 3D printing. आप लोगों ने अगर नहीं ध्यान दिया है तो try to find out. This is an upcoming area. It is actually going to save pharmacy. And then we have patient-specific drugs like semi-solid extrusion 3D printing has been explored as a coating technology for customizing the release route. So all this content is telling that 3D printing has opportunities. It has solutions to many problems which are facing with conventional drug delivery. So if we talk about 3D printing, it can create dosage forms like IR tablet, bilayer tablet, capsule, polypill, sets, implants, and the techniques used are all these which have been listed. The advances made through 3D printing is in personalized medicine, modified delivery, personalized prosthetics, medical devices, all these, and we can use them for treatment of various diseases like cancer, diabetes, CVS, neurodegenerative disorders, and all the possibilities. So 3D printing is also called as additive manufacturing, and it has progressed a lot. 1984 it was introduced as 3D printing, then it became 3D bioprinting, then it came to 4D printing, 4D bioprinting and 5D printing, and now we have 6D printing. तो आज आप 3D printing learn कर रहे हैं, but दुनिया 6D printing पे पहुंच चुकी है already. And what is bioprinting? Bioprinting means when we are using biological inks. जो ink होती है उसकी physical और chemical property, but when we are adding something biological to it, it becomes a bio ink. So 3D printing की problem क्या है? वो एक static design को consider करता है, जो change नहीं हो सकता. जो ये 4D और 5D प्रिंटिंग इसीलिए आए हैं क्योंकि ये स्टैटिक नहीं है, ये डायनेमिक. यू कैन चेंज द डिजाइन इन बिटवीन द प्रिंटिंग, यू कैन चेंज द डिजाइन. दैट इज द ब्यूटी. एंड FD प्रिंटिंग, 4D printing allows accurate imitation of dynamics of the native tissues. 5D printing has potential to fabricate artificial bone for surgery. 6D printing is using smart materials. So this is the slide which is showing the bioprinting for drug delivery. बायो फॉर्मुलेशन में केमिकल, फिजिकल और बायोलॉजिकल प्रॉपर्टीज हैं. एंड दिस इज द प्रिंटर. इट इज जस्ट लाइक अ बॉक्स, यू नो, सिंपल बॉक्स है. आई हैव एक्चुअली सीन इट और उसको वर्क करते भी देखा मैंने. तो 3D बायो प्रिंटिंग भी कर सकते हैं फॉर डर्मल एप्लीकेशंस, ओरल फॉर्मुलेशंस, इम्प्लांट्स, प्रोटीन एंड ऑक्सीजन रिलीजिंग डिलीवरी. लाइकवाइज वी हैव 4D प्रिंटिंग. ये टाइम फैक्टर उसमें इंट्रोड्यूस होगी जिससे कि आप चेंज कर सकते हैं. अगर जरूरत पड़े तो बीच में यू कैन चेंज द प्रिंटिंग प्रोसेस. And 4D printing is used for creating hydrogels, oral formulations, microneedles and implants. So overall, FDA encourages development of complex dosage form and manufacturing processes using science and risk-based approaches. So it has already approved the drug Spiritam. ये tablets, not only tablets दिख रही हैं, but they can disintegrate in seconds. And even we can have personalized supplements, not only the drug delivery system, but supplements, where you can see in this slide, here so I can explain, like we have a combination of vitamins, and they can be designed as immediate release and delayed release both in one capsule. Okay? So that can be possible, but जितने मर्जी combinations करने हैं. So at the end, I want to state this statement: the illiterate of the 21st century will not be those who cannot read and write, but those who cannot learn, unlearn, and relearn. So it is the time when we need to learn new things, unlearn older things, and then relearn them in a new way. Thank you for the patient hearing.
Any questions, please, from the audience?
Ma'am, as you have discussed, like one machine we can prepare a tablet. Now I want to ask, like one machine can put several kind of tablets, but can it prepare some other doses form or single tablet?
Can do anything.
Even microspheres or whatever.
कुछ भी एक ही machine से होगा. I've actually seen the machine working, तो हो जाता है.
So what will be the cost of that machine?
Cost तो आपको देखिए ऐसा है कि depends upon कैसी machine लेना चाहते हैं या उसके parameters हम set करना चाहते हैं. जितना advanced system लेंगे, it's not about machine, you have to purchase a software. तो softwares भी लेने पड़ेंगे ना. पहले वो softwares लेने पड़ेंगे, उसकी भी cost है. Machine किस format की लेना चाहते हैं, आप bio printing करना चाह रहे हैं, 4D printing करना चाह रहे हैं, 5D करना चाह रहे हैं, उसके according cost वैरी करेंगे. तो you can take them to Google for that. आपकी research area interest क्या है, वहाँ पर जाना पड़ेगा.
Important for the coming one simple question from the side of the student, that what is the scope of pharmacists, those who are studying pharmacists, in the field of this 3D printing, when they come out and they learn through that machines and engineering are doing everything, what they will do in the future?
सीखना पड़ेगा. There is no option. That is what the VC sir was telling. कि अगर हमने अपने एटीट्यूड को चेंज नहीं किया, कन्वेंशनली रहे, तो ये कोर्स बंद हो सकता है. वही वो कह रहे थे. तो ये हमें सीखना पड़ेगा. It may be a challenge, but it is not going to work unless and until we change our mindset. करना ही पड़ेगा. We have to understand robotics. We have to understand artificial intelligence. We have to work through it. और उनसे फ्रेंडली होकर ही काम करना पड़ेगा. Yes, AI तो डालना ही पड़ेगा. AI डालना पड़ेगा. जितनी भी AI related elements है, robotics है, सब डालनी पड़ेगी. नहीं तो then तो like for me it doesn't matter if I don't learn, but for them it matters a lot. For them it matters a lot. You have to prepare for the future. Future is not going to manipulate according to you. आपको future के हिसाब से चलना पड़ेगा. उसी के हिसाब से उसको आपको मतलब learn करना पड़ेगा. वही last line थी. Learn, unlearn and relearn. इसीलिए मैंने कहा कि ये सीखना पड़ेगा. At every cost you have to learn.
Any other questions, please, from the audience? Okay. So thank you, ma'am. Thank you. Your words have inspired us and given us a new direction towards the 3D printing. Now it's time to felicitate our ma'am. So I request Dr. Shashi Kiran ma'am and Dr. Ajay Gupta sir to please present shawl and memento to sir. Please, ma'am. very much. A big round of applause. Please, audience, do something at least. Thank you, ma'am. Now moving ahead for our today's next lecture. None other one, that is Dr. Alok Mukherjee sir. He was my teacher when I was doing B.Pharm. All whatever pharmacognosy I know, he was the one who taught me. So before we start, I would like to give a brief description about sir. Dr. Alok Mukherjee sir is a distinguished academician and researcher in the field of pharmaceutical sciences with 28 years of experience. He currently serves as the professor and principal at United Institute of Pharmacy, Prayagraj. Dr. Mukherjee sir holds a PhD from Uttar Pradesh Technical University, Lucknow, along with M.Pharm and B.Pharm degrees from Dr. H.S. Gaur Vishwavidyalaya, Sagar. His contributions to pharmaceutical education and research are extensive with over 77 research publications, 28 international and 49 national papers, more than 24 Scopus Index papers and several books and book chapters published internationally. His academic influence is reflected in his H-Index of 14 and I-10 Index of 21. Dr. Mukherjee sir has actively participated in over 66 national and international conferences, delivering invited talks and serving as a chairperson in various prestigious seminars. He has also played a key role in organizing academic events, including AICT and TCI-sponsored national seminars. As a recognized academic leader, he has contributed to university faculty selection committees, board of studies, and evaluation panels for various institutions, including AKTU, SHORTS, SHU ATS, and Banaras Hindu University, BHU. He has successfully supervised 14 PhD scholars and 56 M.Pharm students. Dr. Mukherjee sir is a life member of professional bodies such as the Indian Society of Pharmacognosy, Association of Pharmaceutical Teachers of India, and International Academy of Physical Sciences. His commitment to academic excellence and pharmaceutical advancements continues to make a significant impact in the field. So give a round of applause for our next lecturer, and sir, my apology if I am mistaken something. Please, sir, over to you.
Good afternoon, all of you. It's a very pleasing morning since 8 March, International Women's Day. I like to appreciate every woman in front of me and in my memory also, because my house is also having one woman with me. So from this stage, I am appreciating each and every woman of International Women's Day. We are here to speak some of the internationally or nationally importance of pharmacy, because we are going through this international conference in pharmaceutical frontiers, especially exploring future trends in technology, pharmaceutical innovations for new era in healthcare. This morning, Dr. Pathak, your Vice Chancellor, has given an insight of some of the importance of pharmacy and the science which we are developing. I am really thankful to all committee members, those who invited me in this August gathering, especially Dr. Ajay Gupta ji, the Organizing Secretary, Dr. Anupriya Madam, Professor Kamala Pathak, the eminent speaker of this conference, Dr. Monica, and other distinguished guests sitting in front of me. And I say especially thanks to some of my colleagues and students, now they become a colleague. And Dr. Shashi Kiran Madam, Dr. Nisha Ma'am, Dr. Meenakshi Gupta Ma'am, Dr. Ajay Yadav, Dr. Ajay Gupta ji, Dr. Anju, Dr. Shweta, Dr. Pratima, they may have gone through a part of my journey of academic, whether they are part of the students or the colleagues in that part of my 30 years of journey of teaching and research. Coming to the theme, I'm basically a pharmacognostic, very ground, natural product part of this pharmacognosy. That's why I came with the theme of this importance of sustainable cultivation and conservation of medicinal plants. Whenever we go for any drugs or medicine, we need to have a certain chemical. We need to have certain chemical compound, and that should be come through apart from some synthetic or biosynthetic or some natural product. And that's why I came for that how this natural source of the drug can be used as a source of a drug. We all know drugs in generally are called poison, but the poison can be very precisely be taken care to become a medicine or a drug. Or if we take in a medicine without any dose or in a prescribed manner, it can become a poison or side effect. So I'm just going through this. We all know drugs are any particle intended for the use of diagnosis, cure, mitigation, treatment and prevention of disease in man and other animals also. We all know, we being a pharmacist, being a pharmacognosy or being a pharmacy students also. But natural drugs are a part of this drug, which are in different sources including plants, animals, minerals or whatever the microorganism we are using as a biotic product. But human being using this potent action of a natural product for a multiple purpose, it may be for directly related to the disease or for the cosmetic purposes or for recreational use patient. And that's why we have to be very careful about these natural drugs, especially when we use as a drugs or the cosmetic also, drug and cosmetic act. Traditional system of medicine in India is basically run by Ayush, which include Ayurveda, Unani, Siddha and Homeopathic system. And this Ayush all way using this natural source of these medicines, whether it is plants, minerals, animals, or whatever they are. The practices of traditional medicine vary greatly from country to country. The drugs which are using by the European countries, the drug using by the US or the China or in India, maybe a therapy will be modified. There are 100 plus or 200 plus different therapies throughout the world they are working. But they all are using a natural sources, different plants and different varieties. That's why the varying in the country to country, region to region also. In India is also, when we are going to the southern area, like in Kerala or Karnataka, the therapy may be different. Or in case of the eastern area or in the central area like UP, MP, the therapy may be modified with the source of these natural drugs. So they can be influenced by the factors such as culture, history, personal attitude and philosophy. And that's why each drug source should be well defined and well creativity through the way of their production as a part of source of the drug. There is a global resurgence in the use of these medicines along with the growing scientific interest in them as a source of new drugs. So when we take that crude drug, it can be designed in a modified way, and that can be a synthetically or biosynthetically can be having a potent drug which can be useful for any treatment or any diagnosis purpose. Herbal drug may vary in the compositions also. One drug which is collected from this area or another area from 100 kilometers from there or 1,000 kilometers from apart can vary in the chemical compound also. And that's why we have to identify what chemical we are looking for, what chemical we are going for, and what type of doses we have to go for. And that's why it is very important to identify the drug and then their varieties and specificity for which we are using for the source of any medicine. So Their use is well established and widely acknowledged to be safe and effective and may be accepted by the national authority. Any drug which we are producing by any method and the methodology, it should be approved through FDA or USFDA or any different authorities throughout the world, and should be pure and potent and having a lesser toxic, should be carefully used by the patient. We all know that throughout the world, and WHO also has reported that 60 to 70 or more than 70 to 80% people are globally rely on the natural sources or these herbal sources. Herbal medicine have made a headlines due to their features and result-oriented therapy and lesser side effects. Although we all know that they are not having a side effect, it is not there. Every drug having a side effect, but this is a lesser side effect than the chemically synthesized modified drugs. The global medicine plant industry is rapidly expanding and has been valued at approximately $160 plus billion in 2022. That's why we are also going towards the different part of the herbal medicine. Phytocompounds and their chemical analogs have provided abundant clinically useful drugs in the treatment of chronic and acute diseases also. And these phytochemical compounds, when we isolate it, very potent compounds. These potent compounds are also being used now in a case of acute and chronic disease also. This growth is fueled by the increasing demand of natural and organic compounds and the rise of chronic diseases and a greater acceptance in traditional medicine, and that is the report of 2023. And this is the scenario. The global herbal market medicine sized around $223 billion in 2024. You can estimate that how the herbal reliance or herbal cosmetics are being used by the every man and woman throughout the world is now used with herbal drugs because they are having a good effect, good potency, and lesser side effect. And that's why every pharmaceutical company have their own natural drug industry also in the field of this area, or in case of India and global area. The market is projected to grow about $251 billion in 2025 to $437 billion in 2032. So the compounding accounting growth rate is 8.23% during this forecast year. You can think about that from 2019 to 2032. The growing of natural sources and natural product market throughout the global is also there. Europe dominates the herbal medicine, but we, especially India and the Chinese medicine, are also a very competitor, and they are growing through with this US or other countries, Europe also. In India also, we are not lacking behind. The CSJR also 6.87%. The herbal product market in India witnessed significant growth recent decade due to the increasing consumer awareness, because we all love or like this type of herbal medicine in our daily use space. The overview of Indian herbal medicine market trend heading towards 2023, and the market is India generated revenue of $4,623 million in 23 and expected to grow about $26,794 million in 2030. And this overall growth is about 28.5% CAGR. This is an exported value by our Ayush department, where all the herbal medicines are being useful, and from 2015 to 2023 they have been exported from $3,308 million to $628 million. So you can see within the last 10 years we are splendidly growing through this market project also. And this is due to that, because China is our good competitor. China is exporting towards the surrounding areas like Vietnam, Singapore or surrounding countries. But we are exporting to the higher nations like US and other Germany countries, European countries. That's why our growth rate is also good. But whatever the market growth, whatever the exponential marketing herbal drugs are there, our aim is to make a quality product. Our aim is to produce a better herbal drug, and that's for we need to know about the quality and the quantity and the cultivation of the drug. Because whatever the green, it is not the drug. Whatever the plant is there may not having a good compound inside. So we need to know what is the drug, how to identify, and where to cultivate, and what chemical compound we are isolating. Unless or until the plant does not contain a good quality or good quantity of that chemical compound, it is worthless. Every green thing cannot be useful. So we need to know what to do with this herbal drug and how to build a better potent drug, which you need to know about this part. So I am focusing on that. What is the role of the cultivation, especially? Because whenever we go for any Indian traditional medicine, we have to identify and select the drug. Then primary screening with a different process are there, we all know. Then phytochemical profiling we can do. We can go for high thoughtful screening also with a different analytical method. Then isolate the drug. Bioactive compound identification by different screening method. You can go for IR, MR or NMR or like this screening, and then come to the market product like after the clinical trial phase 1, 2, 3, 4. But our aim is to go for the cultivation and collection is required. Whatever the farmers, whatever the medicinal plant growers are giving the drugs are not pure or maybe not having a that particular bioactive compound which we are using as a source of the drug. So we need to know how to cultivate the medicinal plants. That is why need of sustainable cultivations are required. Globally, 17 to 18% of blood flora is currently utilized for medicinal purposes. The rest of the things are not being cultivated. And that's why it is our aim to make more and more quality cultivation. Because widely collected herbal plant may not have that much of good potent chemical compound which is useful for the source of medicine. So, यदि हम मान लीजिए कि हम कोई नीम का पत्ता खाना किसी ने बोला, तो कौन सा पार्ट खाएं, कितनी उसमें एक्टिव कंपाउंड है, ये जानना बहुत जरूरी है. कहीं भी नीम का प्लांट उठाया और नीम पत्ती खाना शुरू कर दिया, that is not important. So we need to know whether it is cultivated, whether it is having a good organic compound, good manures are there, whether having a less pesticides or not. This all parameters are to be required, and that is why I just go through some important slides of this cultivation. Cultivation of medicinal plant, which is a key to integrity, quality, efficacy, and safety of Ayush system of medicine by integrating medicinal plant in the farming system, offer an option for crop diversity. What is the role of that farmer? What is the role of that medicinal plant grower? And how they can aware about the plant and the active compound, ensuring long-term availability and reducing environmental impact. That means they should know about what time which plant has to grow, because every season every plant cannot grow, and that is why our aim to decide where to grow and how to grow. So ensuring long-term availability required, because in the market we need every time the drug. It is not that only in winter season this drug is required. So we have continuous supply of the drug, and that means we need to have a cultivation regularly with a proper manner. Pharmaceutical permaculture design. It is holistic design methodology that integrate elements of agriculture, architecture, and ecology to create self-sustaining systems that work with the nature rather than against it. So we have to make a culture, permaculture, where we can derive or design our cultivation with the environment also. So we cannot make against the nature. Plant resilience. The resilient plant can adapt the changing condition and continue to grow and thrive, and even under challenging circumstances. When we grow a plant in unfavorable conditions, the plant itself has modified themselves their phytohormones, their enzymes, their phytoconstituents can modify themselves and get a better and better bioactive compound or biosynthesized synthesized process can be changed inside, and that can be a good potent plant. Promoting ethical sourcing also. Ethical sourcing focus on ensuring that plants are cultivated, harvested and traded in a way that environmentally sustained, socially responsible and economically fair. Because we have to find out whether these plants and cultivated plants can be come to the market in a proper way, and the grower or the plant farmers can get a better and better revenue, so it can economically both ways can be useful for them. So sustainable cultivation of medicinal plants requires knowledge about good collection practice, good agriculture practice and good storage practice. Because once we grow, it cannot be only grow and we cultivate it finished. We have to harvest it in proper time. We have to store it in proper time. Drying process also required. You must have students of our B Farm first year or second year M Farm. They must be knowing the digitals must have to collect at particular time. If it is not collected in a afternoon time, it will degrade the digitoxygen. Why? Because the enzyme itself present in the plant can activate and destroy the digitoxygen some percentage. Because digitoxygen for today's enzyme in the morning, early morning and later evening, it can be have a good effect on the compound. So it is well established that time of collection or harvesting of any plant should be well aware by the farmer or those who are growing the medicinal plant. Similarly, in different plants also, cultivation with a good agriculture collection practice, GSCP, to promote standardization and quality assurance and thereby enhance the acceptability of Ayush system globally and increase export of pharmaceuticals and Ayush products. And that's why day by day, every pharmaceutical company coming another sister person with a natural product or Ayush product that come into the market, and they are delivered a good quality product of natural sources, herbal sources, and that's why every, like in Cipla, in Cadila, in Dr. Reddy, they have a sister concern with the herbal medicine source. For this type of GCP or GACP or GAP, or especially the good agriculture and collection practice, we need to know the practice, sustainable cultivation practice. What is the practice? It mainly focuses on growing crop in that way that it maintains the soil health. Unless the soil health is not good, the plant cannot grow healthy, cannot give a better compound. Conserve water. We need a proper irrigation. The groundwater should be there. Then reduce pollution. We have to maintain that it should be completely pollution-free area where the cultivated medicinal plants are going, and support biodiversity. So this 4 points has to look after when we go for a sustainable cultivation, and this practice should be maintained while cultivation. Similarly, we have to take care of the focusing on avoiding the synthetic fertilizers. We have to go for an organic fertilizer. Pesticide should be reducing, or it should not be used a chemical fertilizer. We are using for the, we should use herbicide or herbal type of fertilizer like nemocide, neems are there. Then genetically modified organisms are there. And this can make our practice to grow a natural source of medicine. We have to manage the soil because soil is a basic requirement. So, soil management techniques need to have a crop rotation. So, whenever we grow the medicinal plant, it reduces the soil depletion and prevent pest build-up. So, pest should be lesser and the depletion of soil will be lesser. The rotation of the crop can be done. Compost and green manure required to improve the better quality of the product, and soil fertility should be given. Mulch retain moisture and suppresses weed and regulate temperature. This is also one part of the practice of good medicinal cultivation. No till farming reduces soil erosion and maintains soil structure. So we have to maintain the soil, we have to maintain the water, and rotation of the crop is a very important part. Water conservation is also a part of organic farming, where we have to make a drip irrigation, rainwater harvesting, then cover crops also that can maintain a better quality of medicinal system of cultivation of medicinal plants, which prevents the water runoff and improves the soil moisture retention much more than. So we have to be very careful about the cultivation when it is being a part of growing the plant, medicinal plant, in a soil which is effective soil fertility. This is a comparison of the crop rotation. We can use a mixed cropping and crop rotation. Soil fertility improved by crop rotation, while in case of mixed cropping, continuous nutrition utilization. Soil pest control is also reduced in both ways, but there are some merits and demerits are there. But major challenges is there. We have to aware the people, those who are working in the cultivation of this and the conservation of the medicinal plant. If the people from the ground, the farmer, not being aware, not having a good proper knowledge of growing a medicinal plant, we can get a plant without any medicine compound or having much more pesticides inside. So they are just a waste of the product, because our aim is to get the potent compound with a lesser toxic value, and that is why. So growing a cultivation of the medicinal plant is very important, and the knowledge awareness for the farmers is also important. For that, National Medicinal Plant Board is there, NMPB. What it gives, the National Medicinal Plant Board, it is derived by the central government, or also the state governments are also there. They design a comprehensive support system that not only ensuring the availability of the quality medicinal plant, but also enhances the livelihood of farmers and support teams and aware them what are the importance of the medicinal plant and its value. What they are giving training and capacity building to train them and giving the better and better knowledge of harvesting, their agriculture practice, the management and the marketing also. Financial assisting for the nursery maintenance. These national boards will also give the farmers to how to grow the product and how to market the product. Like lot of people are extracting the oil, essential oil, but they don't know how to sell it, where to sell it. So the crude oil can be refined oil in a market, like we are using a different type of deodorant with a different type of oil for our refreshment. But these drugs will be marketed in which area and how does the market has to be go through, and that's why these National Medicinal Plant Board are giving the awareness to the farm. Information, education and communication activities are regularly growing with this board. Species specific campaign, that means variety of the different species of the same plant has to be knowledgeable. While working in the South in India, the Kerala people, हम जानते हैं कि Kerala is a part of the state where the spices are very good. But in India, in different other states, we can also grow this type of species. So species specific campaign is a requirement. Board launches campaign for a specific medicinal plant, their knowledge and their usage in planting also. Marketing support, then minimum support price for their livelihood. The farmers should have at least get the MSP. For that, they have to be a part of this. They are giving the institutional extending also. They are giving the guidelines to the institutional which are having involving in this type of harvesting, cultivation, or the enforcement of this part of courses are there, where the State Medical Plant Board has also governed the institutional, especially CGs. The conservation studies are especially two types: effective in situ or effective ex situ conservation. So the plant after growing, but they are technically modified with a high quality and higher quantity of medicinal compounds are required. So protecting medicinal plants within their natural habitat, this approach maintains the ecology, its processes and genetically diversity of the plant population. So growing in a field without any knowledge, it is better to cultivate with a proper knowledge that gives you a better and better plant product. Similarly, the ex situ conservation, the preserving plants outside their natural environment, such as botanical garden, seed bank, so the controlled cultivation. This method safeguards species from habitat loss and allows the researchers and education for the better plant in the medicinal field. Biotechnology also helps it because this ex situ, in vitro or in situ are in the part of where we can propagate micropropagation by the biotechnology technology, which we have studied some of the part of this syllabus in that case, where we can use this technology for getting a better quality like dairy root culture, the plant modifications, then incorporation of some biosynthetic product, bioprecursors we can be used, and that can modify the plant product, or the toxic compound can be researched, or the potency of the particular compound can be enhanced. So in vitro conservation of genetic material can also be modified by biotechnological process. Genetic improvement and the breeding is also there, by which we can modify it. Biotechnological enables the genetic modification in the medicinal plant to enhance their resistance to disease, pest and environmental sins. And this gives you a resistant plant which can be away from the different type of fungal or insectal disease or species. Conservation of endangered species, we can save few endangered species, especially we have gone through some vaccination plants, vaccines from where the plant can be, the vaccine product can be collected from the plant, and anti-malarial drugs, resistant anti-malarial drug, it can be out from the endangered plants. So these plants are being, again, can be developed by this biotechnologically, and this drugs are being available for the people who are resistant from malaria. Phytochemical profiling are there. We all studied all these things. Biotechnology can enable the identification and characterization of chemical profiles of medicinal plants, and scientists can ensure that the plants with the most potent medicinal compounds are conserved for the future people or future. Certain challenges are also there where we have to have cultivation for that, over-exploitation, lack of suitable technology, lack of skilled workers being required, and loss of traditional knowledge. So these are the challenges for the medicinal plant cultivation we have to look after, and we have to grow through a better and better, proper, knowledgeable persons, by which we can have different challenges for market price fluctuation, habitat degradation, biopiracy, and lack of regulation and infrastructure. So we have to provide the farmers or the medicinal plant growers or the cultivator for a better environment to grow up good. Now, just coming for this theme of future trends of medicinal cultivation, which require sustainable farming practices that I discussed, the climate smart farming, resource efficient farming, and tissue culture technique, biotechnology technical, plant tissue culture, dairy root culture is required. Genetic engineering is required as a future trend in medicinal cultivation. Then comes the artificial intelligence, data analysis. That means we can identify the disease before it can be treated in a plant. So different disease identified by the artificial intelligence. Nutrient deficiency detection. That means plant is growing through, but whether the active compound is having a good quality or not, we can derive or we can detect from the AI process. Similarly, the growing monitoring, whether the plant is growing better or not. The whole plant is growing, but we need a flower, we need a fruit, but the fruit is growing through or not, whether it is having a good compound or not, that can be also identified this artificial intelligence, that we are having a good stress detection. The plant, if I am having not good irrigation, that means the stress compound is going through. The total biosynthetic modifications are required. We need a A compound. Now, due to the stress condition, it becomes an A1 compound. It is what? We need a aldehyde compound. It becomes a ketonic compound. So again, we can identify by AI while going through the plant compound. Irrigation optimization, yield protection we can develop by the AI, and these are the future trends required for the cultivation of the medicinal plant. So I am just concluding with that. The sustainable cultivation of medicinal plants signifies consumer demand and stressing the importance of agrochemical or agroecological methods, while conservation, initiation, promoting an industry's long-term survival while preserving natural resources and promoting economic prosperity. The sustainability produces a closed-loop system, a circular economy, repurposing of agricultural byproducts. That is very important nowadays. We can repurposing the agricultural byproducts, which are generally we are not using nowadays. Reduce waste, reuse and recycle resources. That is more important. How to reuse the product which are byproduct, and we can make it in a different type of wrapping product or the product which are used for the packaging product also. So it signifies enhances the values of raw materials, creating profitable opportunities in the pharmaceutical industries and cosmetic industries, especially what we are using for drug and cosmetic act. And this includes biological active compounds like alkaloid, terpenoid, flavonoid, and different phenolic compounds which are useful in our mankind daily life. If it is a quality product, then it will be good for us. So cultivation for the medicinal plant is most important. These are cultivation. You can make it better and better. Thank you for conserving the medicinal plant. Thank you. Any questions?
Thank you, sir. Any questions from the audience? Sure, please.
Definitely, this presentation was demand of time, and you were very nicely awakened the audience. My very little small question is here because I am also interested in this field. What is the major solution of the over exploitation? How we can prevent it?
That when we have a lesser knowledge how to produce a plant, how to produce a medicinal plant, because once we are not having a knowledge of what chemical compound we are looking for, we are just exploiting the plant growing and take it away. But we don't know whether the plant having that particular compound, that chemical compound has been modified or not. Like if you go through a simple coriander, when it is a unripe coriander, it does not contain any flavor, any terpene. It having a long chain, trans-tridecimal, decanol. But once it become a ripe product, धनिया जब ripe हो जाता है, तब वो cyclic compound बनता है. Then it becomes a coriander, and it is having a good smell of dhania. So once we have not knowledge of what chemical synthesis is going through inside the body as a biosynthetic product, so over exploitation is there, that when we are growing the plant and harvesting the plant without any knowledge and exploiting the market without having a good medicinal potent plant in a market. Thank you. अब एक आंसर आपसे बता सकते हैं, जैसे अभी शिवरात्रि गई, धतूरा का प्लांट बहुत कॉमनली आपने देखा होगा, बट इट इज ए वेल पोटेंट एट्रोपिन कंपाउंड. लेकिन एट्रोपिन क्या धतूरा में होता है? नहीं, धतूरा में एट्रोपिन होता ही नहीं, बिकॉज इट इज अ रेसमिक कंपाउंड. तो रेसमिक कंपाउंड प्लांट नहीं प्रोड्यूस कर सकता. प्लांट केवल प्रोड्यूस करता है उसके आर और एल और एस टाइप के मॉडिफिकेशन. उसका इक्विमोलर कंपाउंड जब मिक्स करेंगे तो एट्रोपिन बनेगा. और एट्रोपिन कितना पोटेंट है आप सब जानते हैं, क्योंकि आई के डायग्नोस्टिक में यूज होता है और प्री ऑपरेटिव होता है. सिमिलरली फर्स्ट स्लाइड आई गोन थ्रू दैट बेल जो यूज करते शिवरात्रि में हम लोग यूज करते हैं, अभी रिसेंटली गई, बेल का क्या प्रोडक्ट है. सो दिस टाइप ऑफ नॉलेज इज रिक्वायर्ड. It's a weedy plant, natural plant growing everywhere, but we have to know about how to cultivate in proper way to get a better potent compound. Thank you. Any other questions?
Any other questions, please? Thank you, sir. Thank you for sharing your insight and experiences. and sparing your valuable time for us. Now it's time to felicitate Dr. Alok Mukherjee sir. So for that, I would like to invite Dr. Prakash Chand sir and Dr. Nisha ma'am to please felicitate sir by presenting shawl and memento to sir. Please sir, please come. Audience, please have a round of applause. Thank you, sir. Thank you, ma'am. Now we proceed further for our next lecture by Dr. Monica Arora, ma'am. She is from Maldives. So, ma'am, please come. And before she starts, I would like to give a brief description about Monica, ma'am. So, Dr. Monica Arora is an accomplished pharmaceutical scientist and educator with an extensive experience in academia and research. She is currently working as an assistant professor at Villa College, Maldives, and previously served as professor and head in charge at Alameen College of Pharmacy, Bangalore. With a career spanning over 18 years, she has contributed significantly to pharmaceutical chemistry, drug design, and medicinal research. Dr. Monica Arora earned her PhD in pharmaceutical sciences from PES College of Pharmacy, Rajiv Gandhi University of Health Sciences, Bangalore. Her research interests encompass computational drug design, molecular docking, synthesis of bioactive compounds, and pharmaceutical analysis. She has successfully supervised multiple postgraduate and undergraduate research projects and has been the recipient of competitive research grant for innovative drug discovery projects. Her scholarly contributions include numerous research publications, book chapters, and invited lectures at national and international platforms. She has actively participated in academic conferences, faculty development programs, and professional workshops, while also playing a key role in organizing scientific events. Dr. Arora is a recognized academic reviewer, examiner, and member of faculty selection committees. She is a lifetime member of Indian Pharmaceutical Association and the Association of Pharmacy Teachers of India, demonstrating her commitment to advancing pharmaceutical education and research. So we welcome you, ma'am, to deliver the lecture. Please, audience, have a round of applause for ma'am. Over to you, ma'am.
So good afternoon to everyone. First of all, I would like to thank organizing committee, Dr. Ajay sir, Dr. Anupriya madam, and other members of organizing committee for inviting me and sharing my thoughts on this topic. Secondly, I would like to extend warm greetings on Women's Day, as we all are, our population over here is maximum women are there. So I would extend my warm greetings on Women's Day, and I would like to cherish the small thoughts what we all have to grow as women, and I really cherish when we come out of the home, taking care of the family, coming at the workplace. So I would like to share a small thought process of mine. Let us grow a bit more at our workplaces, because that is the need of the hour. So as today is International Women's Day, so let cherish all the women around us who contribute at their workplace, and at the same time they are taking care of the families, coming out. So yes, so with that thought, I will be starting my presentation. So the topic which I have chosen is, as per the panel, what they have given me the topic, that is the role of the personalized medicine. So when we talk about personalized medicine, this is one of the healthcare delivery system which is really coming in, which is upcoming healthcare delivery system. So when we talk about that, we talk about the word personalized. So personalized means individual. So when we talk about individual, they will be having their own genetic setup, they will be having their own environmental setup, as well as they will be living at certain geographical location with a certain lifestyle. So when a certain individual will be having all this aspect, of course they will be having over this period span of time, they will be having certain disease aspect also. Now looking into those aspects, what kind of healthcare delivery system we are that is upcoming is the personalized medicine system. So let's see what's going ahead. So in this presentation, we are going to have an introduction, the various core principles, applications in healthcare, then what will be the role of pharmacists, and then of course what challenges we are going to face while it is one of the implementation aspect also. Then what is the future of the personalized medicine, and then the $1 million question means the conclusion, and of course we'll be having certain references. So as we talk about the term personalized medicine, it is also known as, coined as a term, precision medicine. So basically it is an approach of the medical treatment and that prevention that comes into account while we talk about their individual variability, we talk about their genes, their environment, their lifestyle. Basically, whenever we fall sick, what happens? We go to a doctor and the doctor diagnoses, they prescribe. So it is a traditional one-fit approach. That whosoever is going, doctor is diagnosing in the same way, they are prescribing the same medication, without considering that what are the various other factors that has to be looked upon. One of the important factors is the genetic factor, which we hardly take into the consideration. But in this personalized medication approach, we first look into the genetic aspect of the person. Now, when I say the genetic aspect means, whenever personalized medication come into the picture, the individual person, because it is an individual approach, the individual person has to undergo a genetic counseling. Genetic counseling is a counseling that has been done by a specific healthcare person. They take the particular person genetic aspect, their lifestyle, their environment, and they predict that they will be having in their lifestyle, they may have certain lifestyle diseases, which we very well know are very alarming these days, which involve diabetes, which involve cancer. So eventually they will be tailoring their medical care aspect. So as the term tailoring means, as per their individual requirement, as per their genetic setup, they will be tailoring their treatment and then whatever other intervention also, which will be specifically characteristic to the patient. So now we can very well see this is a one-fit approach, that all the population, we undergo the same process of healthcare system. Now in this particular newer healthcare system, it is this is a conventional healthcare system which we are following, but in the personalized medicinal approach, we are basically treating based on the patient's unique genetic profile. Now to know the unique genetic profile, we have to make sure that the person undergo the genetic checkup. So once we have their genetic profile, so eventually we will be giving them the targeted therapy or the satisfied therapies. Now, these are the certain aspects of the personalized medication. The most important one is the genomic sequences as the foundation. So, any individual we want them, they should take the personalized medication as a healthcare delivery system. They will be having a detailed analysis of their individual genetic composition, which happens through the genomic sequences, that allows us for a deeper understanding of genetic variation. that of course influence their health and as well as the disease. So in the precision as a treatment, so our goal is to deliver the right medication as well as the right doses at the right time. Now we may add that we have been doing it in the one fit also, but no, in the precision as we are knowing the genetic profile. So now by the advancement we can know to that extent that at what age, because we will be having certain biomarkers, will be getting to know at what age the patient will be susceptible to certain disease. So when we know that patient will be having a certain disease, so rather we start treating them in a preventive way. We don't treat them when they fall sick. We start treating in a way because of their genetic biomarkers. We are aware that this is the condition a patient may have over the period of time, and this only happens when this genetic counseling also happens time to time, because over the period of time this genetic counseling may also appear in a certain different way. So that then in this process enhanced diagnostic accuracy also plays a very, very vital role. Once diagnostic is proper, eventually the tailoring aspect will also become proper, and that also allows us to early detect the disease, which we can timely intervene, and then eventually we can improve the patient outcome also. So this particular approach allows us to empower our healthcare system. So the approach is the preventive healthcare system. It's not the treatment approach. It is the preventive approach. As we know the genetic sequence of the patient, of the individual, eventually we will be knowing the various biomarkers which are responsible for the certain diseases. Now, on what core principle that we say we are, claiming that this is in the upcoming days, or rather in these days, it is going to be one of the efficient healthcare delivery systems. So, the most important core principle is the precision diagnosis. Means we are going to diagnose the disease at a very early stage. At the same time, we are diagnosing with the precision. This is only happening because we have the genetic profile of the person. So once we have a strong genetic profile, we are having their biomarkers. With the help of these biomarkers, at the early stage only, we are diagnosing, and eventually we are making a tailored treatment plan for them, which is leading to more focus and the effective treatments. So because of this precision diagnostic, another pillar is the customized treatment. As per the profile of the patient, we are tailoring their treatment plan. So we are going to select the treatment which are based on their individual likelihood of, and as well as the effectiveness of that particular medication. Now this customized treatment plan, as it is a preventive approach, that reduces the risk. Because we know the biomarkers, we will ask the patient to change their lifestyle approach. So as whatever disease we are having, we are all aware that they are kind of a lifestyle disease, whether you say diabetes, cancer, so they are very alarming disease these days. So when we know that a patient is susceptible, so we eventually ask them to change their lifestyle, and eventually when they change their lifestyle, they will be having certain less amount of impact, that they may have less chances of having this disease. Eventually they may not fall sick. Now the most important aspect is the genomic sequences, which is the foundation of the personalized medication. In this, the patient undergoes a detailed analysis of the individual genetic composition through genomic sequences. This allows us for a deeper understanding of the genetic variation that influences the health and the diseases. So under this, the precision in the treatment happens to deliver the right medication at the right doses at the right time, and then eventually the patient will be having enhanced diagnostic accuracy, and then eventually the empowering preventive healthcare comes into the picture. So this is again one of the major core, that is the preventive care, which is the personalized prevention, that we are working on to the prevention aspect. Then when we work on the prevention aspect, the patient undergoes the holistic approach. When we say the holistic approach, we have to make awareness regarding the lifestyle adjustment, various screening methods that allow the patient to know that they will be having certain biomarkers, because of that they may fall ill, and then the early intervention to reduce disease risk and the improve well-being. And then of course innovation in research also comes into the picture, because of the biomarkers available. We will be aware that what kind of the disease the population may be having over the period of time. And then looking to that, we can have the targeted intervention. We can focus onto the molecular pathway, which create more effective and the various personalized treatments. Now, what does the personalized medicine promises us? So basically, it promises us about the genetic information, which often involves analyzing genetic profile to understand how their gene influences their health, various disease risk and response to the treatment. Looking to that, once having their genetic profile, a tailored treatment plan can happen, and then which provides the developed personalized treatment plan, which are more effective, and they are very much potentially safer than the generic treatments. Looking to the personalized medication aspect, various preventive strategies can also be planned that doesn't allow the patient to fall sick. We will be getting to know their unique risk factors, because we are aware of their genetic predisposition of certain diseases and other lifestyle factors that may increase their risk. Looking to that, what can be done is that all the preventive strategies, tailored treatment plan, the doses form which we are using in the personalized medication can be monitored and adjusted. This could further include the regular genetic testing, which involves the monitoring of the various biomarkers. Now, we have been talking about the personalized medication. The most important role in this healthcare delivery system is played by the role of the genomics. The reason for this is that because we are going to have an understanding and analysis of the various genes that will allow us to understand predicting the disease risk. Looking to that, the genomic analysis allows us to predict the disease, which enables the individual to choose various prevention plans that suit their lifestyle. Then we can also optimize the drug selection. Genomic also plays a very, very important and crucial role in selecting the right drug at the right time, ensuring the optimal treatment that outcomes and minimizes the various adverse effects. And then of course we have the personalized prevention. This allows us, genomic information allows us to develop a personalized prevention strategies, reducing the likelihood of developing the certain diseases. Now, we have been talking about this, and of course when we talk about the personalized treatment, we talk about the collecting the genetic data and using them as information for various, identifying the various markers. The artificial intelligence plays a very, very vital role, because it allows us to collect the data of the population, and eventually when we collect the data, artificial intelligence allows us to ensure that a personalized care plan can be made with the help of that. And then it also helps us to allows us to analyze the vast amount of the genetic data to identify the various patterns, and which help us to predict the medication which can be most effective for a certain patient. Then of course, as we are working on the preventive aspect, we can smartly monitor their biomarkers, and if we find that there are certain changes, what we can do, the healthcare professional can take care of their aspect. They can plan their lifestyle, they can plan their treatment strategy. And then of course we have the data-driven insight, which AI can integrate genomic data with the other healthcare information to create a comprehensive patient profile, enabling more information treatment decisions. Then what role, being pharmacists, we all of us have here? The role which we are going to play in this delivery system is first that we should be first of all aware of this kind of healthcare delivery system, which soon it is going to be implemented. We have to ensure that we are going to, of course, taking care of their medication management, and then in this we will be taking care of their personalized medication aspect. we will be helping the patient to understand how does the tailor medication work on their genetic makeup, their medical history and other individual factors. Eventually, the pharmacists will also be helping us in the various adjustment of the dosage form and the various formulation. And then the most important aspect will be, as it is based on the genetics, the genetic counseling. Probably we are not much aware of this, but soon as this delivery system is going to be get implemented, being pharmacists will be having more understanding how to do the genetic counseling and how it will be, how we have to ensure that how are we going to learn it even. So in this, we are going to see that genetic testing support, and of course, to have a genetic profile, we have to do a genetic testing of the patient. So first thing is that we have to, will be quite vigilant enough that how to do the genetic testing, and then once we do the genetic testing, eventually we will be having results also. So how to understand those genetic results ourselves, and then how to ensure that the patient whom we are assisting to understand it, and rather than getting afraid of it, understanding that it is for their better compliance. Then next is, after the understanding, the interpretation of the genetic data. We may be having a certain genetic data. First of all, we have to understand the genetic aspect. Because of this reason, we should have an expertise in the genetics. Then only we will be able to interpret the genetic data. To make this process in a smoother way, there has to be a collaboration with various healthcare teams which effectively integrate the genetic information into the personalized treatment plans. Now, next aspect of the pharmacist is to have an understanding towards the drug reaction management. They will be, because of the genetic counseling, they will be a better place to recognize and then to manage the adverse drug reaction related to genetic variation or to the individual factors. So, pharmacists are also going to play a very pivotal role in mitigating the potential risk associated with the personalized medicine. Then, of course, the patient education and counseling is the most important parameter, because to have a change is not easy. So, because patient will not be aware that what is going to happen, why are we even doing that? So the patient education and counseling is very, very important, that how this genetic counseling will work, how the medication they are having priorly to prevent this particular diseases, what potential side effects they will be having, and the various, and then the importance of the adherence, because they may not have the disease. So we are treating in a way that they will not have disease, but they have to take probably some certain supplements, or they have to do certain lifestyle changes, which they will not be able to understand, and then to adhere it itself becomes a difficult task. So here, the pharmacists play a very, very important role to make them understand that this is because this is all is happening, so that rather they don't have a disease also. And then eventually, over the period of time, if they have also, as we are aware, so we will be already having a tailored plan with us. Then we have to educate that how this tailored plan is going to work, and why this genetic matter is influencing the various treatment response. So they will be having certain doubts, certain questions, which they will not be understanding. As we very well know, when we being pharmacists, anyone come and talk about medication, it is difficult to make them understand. And now we're talking about we have to make them understand with the genetic aspect. Of course, it was going to be a tedious task for the pharmacist also. So in this, the collaboration with healthcare team is going to give them a beneficial result. Here, the pharmacists have to work along with the physicians, genetic counselor, and the various specialists to develop the comprehensive personalized treatment plan. Now, being pharmacists, we are also involved in the manufacturing and other research aspects also, and we will be, as we are going to have a genetic data of the population. So when I say that term population means the masses. So we will be having an idea that the population is moving towards which kind of the lifestyle diseases, what diseases are going to be prevalent over the period of time. So eventually, the research can lead to in that year, rather doing research with we are doing, that may happen. But because of this personalized medication is aspect, the research will have a direction that the particular research will be happening regarding to certain diseases which is going to, which may be prevalent over the period of time. Then of course this is going to transform the entire pharmaceutical industry. For example, we'll be having a targeted drug development. We'll be doing the development of the drug in a way that that particular we are knowing that certain diseases are going to be prevalent. So eventually, the pharmacists, the researchers will be focusing more onto the target drug development, and then faster drug discovery. That, as we very well know, that this drug discovery itself is a tedious process. But when because of the personalized medication aspect, we are aware that these are the lifestyle diseases which the population is going to face in 15 years or 20 years. So eventually, the drug discovery will also become faster. And then the precision medication initiative, to initiate it, first we have to initiate the precision medication, which of course to initiate something and to implement something really takes time. Now these are the various applications of the personalized medication: diagnosis and intervention. It enables us to more precisely diagnose and the tailored treatment plan based on the patient's unique genetic makeup. Then second is the drug development. Next is the theranostic, that is a newer approach with the help of the personalized medication, which combines the diagnostic and the therapeutics, which uses similar molecules for the both diagnostic and the treatment, particularly in the field of the nuclear medicine. Further, cancer genomics, that is one of the prevalent, very much prevalent condition across the globe. Cancer genomics, it helps us to transform cancer treatment by identifying the specific genetic mutation that can be targeted with the tailored therapies. Cystic fibrosis, it also helps us to development of the drug that targets specific faulty CFTR proteins, which improves the lung function in patients with the cystic fibrosis. And then COVID-19, this also allows us to identify the high-risk genes which are responsible to COVID-19 and certain kind of viral diseases, which also help us to develop the targeted treatment and improve the patient outcome. Now we have been talking that yes, this is one of the upcoming healthcare system is personalized medication is truly personalized. My view over this is that if you remember when I started my presentation, I told it is an individual approach. But actually, when we say that an individual approach, every drug what we are giving to an individual has to be taken care based on their genetic nature. But actually, what we are doing is we are giving the drug, we are marking the population that okay, fine, 35 to 45 of age may face this kind of a diseases. So we are rather not marking it individually, we are giving drug to a subpopulation. So the example is trastuzumab in the breast cancer treatment, HER2 positive, which were receiving this particular drug. So we have not approached them individually. We are giving that because of the certain biomarker present, we are giving that drug to all the subpopulations. So still there is a long way to go in personalized medicine. So now what are the various key challenges which we are going to face? First one is the data privacy and the security concern. Of course, this is the first and the foremost because we are having a genetic profile of the population which we are studying. So eventually we are having their data that we are getting to know all their biomarkers, when disease, what time they will be having a certain lifestyle disease. So it becomes a very sensitive issue, and then if we have the data also, are we in a situation to protect it? So that is one of the biggest concerns when we talk about the personalized medicine. And then of course we have the high cost associated with genetic testing and personalized therapies. So when we know that healthcare is not a very affordable system. But when we talk about one step ahead of it, eventually the affordability becomes more difficult. So first of all, a patient has to undergo the genetic testing. So if a doctor asks us to go for one more test, probably we resist. But when we ask with the help of personalized medicine, we have this genetic approach, it becomes really difficult to convince, and of course affordability is one of the matter also. And then one more challenge is to collaborate between the interdisciplinary healthcare sectors. See, we all are working in different healthcare sector, doctors, pharmacists, nursing. So when we talk about one kind of healthcare delivery system, then there has to be a proper collaboration with all the among healthcare sector, which is a real key challenge when we talk about the personalized medication. But now the question is the personalized medication is going to be the future? So yes, it is going to be the future in abroad, in US, in UK. It has been already implemented. But there the one fit approach doesn't work. The doctor, as soon as you go to a doctor, they will ask you to undergo a genetic counseling. So they will be getting to know the various biomarkers, then they start their treatment plan. And of course, this is going to stay with us over the period of time. The reason being is that now the kind of lifestyle diseases we are having, our population is facing across the globe, it is a high time to think about of something which helps us to prevent, rather than going for the treatment. This particular approach of healthcare delivery system allows us to understand why are we getting diseases, what can we do that we should not have the diseases. So because of this positive approach towards this healthcare system, the personalized medication is going to stay with us. Only thing is that there are certain huge challenges which being pharmacists, being part of the healthcare system, over the period of time we will be in a better place to overcome it. So these were the references which I followed. And thank you so much for the listening, and I know this is a very new approach. So I would like to answer if you have any questions.
Thank you, ma'am. Any questions from the audience, please?
One question. Ma'am, how can we make it affordable for a public?
Affordable for the public, yes, as it is in a very initial state. So what happens whenever anything which comes initially, of course the cost will be high, but as it starts getting popular and people get good response, eventually that particular aspect will affordability starts coming in a better way. So we hope that of course right now it's a costly thing, but over the period of time, and as it is becoming very popular, and in India also, if you go to metro city, there it is very popular. People, and first of all, the awareness. More you are aware, then eventually you will not want to have diseases. Then you will, have a you will reach out to doctors. You talk about this. Then over the period of time, it may become affordable.
I would like to add something. Sure. If you are aware, then you might be knowing that recent products in the form of a smartwatch that will operate, that is equipped with the AI, and that will operate on the basis of your DNA makeup. That will assist the physician, and that is already there in the market. Of course, the cost is a little high, but it is introduced in India.
It is introduced.
Some more questions? Any queries? Okay, thank you. Thank you, ma'am. Thank you very much. Now it's time to felicitate ma'am. So for that, I would like to request Director of Pharmacy, Shashi ma'am, and Ajay Gupta sir. Please come on stage, sir and ma'am, and present shawl and memento as a token of gesture and gratitude from School of Pharmaceutical Sciences. Thank you, ma'am. Thank you, sir. And thank you, ma'am, for sparing your valuable time. So we are grateful to our esteemed speakers and panelists for sharing their knowledge, insight and expertise. Your words have inspired and given us a new direction to explore future opportunities regarding new innovations. So now it's time for lunch. And one more information or instructions for the participants of oral and poster presentees, that they have to reach at their the prescribed places at 1:30 to place their posters. Okay, so I think our hospitality committee will guide them. So now we can proceed for lunch, and for participants the lunch has been arranged at the ground floor, and for the dignitaries our hospitality committee will guide them. So please proceed for lunch, and we will again gather here at 3:30 for valedictory functions. Okay, so all the best for your oral and poster competitions, and please I humbly ask our hospitality committee to escort our guests for the same.